Good morning, everyone. We are going to wait for around 5 to 10 minutes, 5 minutes more, so that all the participants join the meeting, and then we will start with the today's program.
Hello.
Good morning, everyone. So we are going to start with the session. We are still waiting for some of the participants to join, but in the meantime, we can already start with the session. So good morning, all who are on behalf of SPPM NMIMS SPPSPTM. I, Dr. Patel, welcome you all for the workshop, Recent Trends in Drone Visibility Techniques. Before we start with our workshop, I would like to give a brief introduction about our school. So our school, SVKM, NMIMS, SPP, SPTM, is a part of the Sri Vileparle Kirwani Mandal, which was established in 1934. And later on, the Narsimondri Institute of Management Studies was established in 1981, which became university in 2003. Today, we have more than 17,000 students as a part of the NMIMS at 8 different campuses, which has around 17 schools. In our NMIMS University is MAC accredited with A plus ranking, and it's a tier one university. Our school, the SPP SPTM, Shobhaben Pratapbhai Patel School of Pharmacy and Technology Management, was established in 2006. And later on, another pharmacy school, SPTM Shirpur, was established in 2007, followed by SPTM Hyderabad, which was recently opened in 2019. Uh, there are different programs which are offered at our school, starting from the PhD in pharmaceutical sciences, Master of Pharmacy in 4 different disciplines: pharmaceutics, pharmaceutical quality assurance, pharmaceutical technology, and industrial pharmacy. And also the postgraduate and graduate courses are offered in BPharm plus MBA Pharmatech, and as well as BPharm, and the DPharm course is also offered at our school. Our school has different approvals or accreditation from PCI, NBA, as well as AICTE for different courses which are offered at our school. Our school has excellent infrastructures. We have very highly needed the facilities available for the research. We have two cell culture facilities. We have a central instrumentation lab. We have a approved animal house. We have a medicinal garden as well as the pilot plant. One of the important facility which is available at our school is the LC-MS instrument, which is outsourced for the for the outside sample analysis as well. Uh, our school our faculty has been successful to publish several papers in international journals and national journals. There have been several patents to the name of the faculties. There are around 4 patents which are granted and 35 patents which are filed and are in the process. Our faculty have also received several research grants from government institutes as well as industry, which amounts to more than 883 lakhs, which through these grants, we were able to hire around 20 junior research fellows from university, government of India, as well as the industry grants. There are several awards and honors or achievements uh, for our school. Our school was ranked 13 among all the pharmacy colleges in India by the NIRF ranking 2020. Our school has also received platinum ranking in the AICTE CII survey of industry-linked technical institutes 2019. Our school was also awarded as best school in pharmacy by Stars of the Industry Award. So with this brief introduction about our school and university, I would like to just brief you about the schedule of the day one. We are going to start with the first session by Dr. Mayur Yergiri, who is a professor and head of the Department of Pharmaceutical Chemistry at SPPSPTM, SVKM, NMIMS. He is going to talk about the drug discovery post-COVID era. So he will brief you about the different policy changes and what is the importance of drug discovery post-COVID era. The next session would be X-ray crystallography in drug discovery, and that session would be conducted by Dr. Vijay Thiruvenkatam, who is assistant professor at IIT Gandhinagar. This session would followed by a lunch break, and then I would be demonstrating some of the softwares which would be useful for the drug discovery. In the next session, in the tomorrow's session, you will be having a demonstration on the use of uh, the docking software. So I would try to create a base for tomorrow's session so that you will be able to follow the demonstration which will be given by Dr. Ashish tomorrow. So with this, let us start with the first session. But before we start with the first session by Dr. Mayuragiri, let me briefly introduce him. Our first speaker is Dr. Mayuragiri, who is head of Department of Pharmaceutical Chemistry. He is an expert in the medicinal chemistry and drug discovery. His expertise is in the multi-drug resistance in cancer. He has worked for last several years on the acridone molecules, and based on his work, he has received several research grants from government of India. His work has been published in several reputed journals, as well as reputed journal, and he was able to receive several grants based on his earlier work. He is very much expert in the drug discovery, and he is going to brief us all about the drug discovery in the post-COVID era. So I would like to now ask Dr. Mayur Regiri to start the session. Thank you, sir, for joining us and request you to start the session.
Okay, thank you, Kapil. Thank you very much for the introduction. So, at the outset, I welcome all for this uh, workshop because we were possible to do this because there was some money left with the DST Serve grant. So we said that we should utilize this and encourage our youngsters to take up the field of medicinal chemistry because the situation has changed drastically in the post-COVID era. So, at the outset, I would thank our Honorable Chancellor, our mentor, Dr. Gandhi, and our Dean Bala Madam because she couldn't make it because of her busy schedule. But anyway, we will carry forward this. And I also have to thank my colleagues of the Farm Chem department, especially Kapil, Ashish, and Neethal, and then Mayank for all involving in this aspects because and let's begin with something. I'm not going to talk about a lot of research, but I will give you a brief inputs that what has happened in the last 1 year or during this lockdown period. I mean, things have changed drastically. So I will give you some inputs because I do make some policies for the government. So I will also share with you what recommendations were made, and some recommendations couldn't be taken up because positive, basically due to paucity of funds, because in the COVID era, because you know that our budget has come down drastically, and but thank God in the coming budget there's a huge impetus and huge money put into research. That's a welcome thing for us. So is my slides visible?
Yes.
Yeah. Okay, good morning, everybody, and welcome all the participants and the registrants for this workshop. So I will brief you some small things which you may take up, and I would because I have seen a lot of people not taking medchem in the masters, and I would rather encourage them to take it up because things have changed in the post-COVID era. Because I'll brief you some of the things that are happening, and then if you have any questions, I can answer you. So I will not bore you much because there's not much slides also. What I can tell you is in the post-COVID era, the number of demand for medicinal chemist scientists has increased drastically because I use LinkedIn and I have seen across the world, especially Europe and US, there is a huge demand for medicinal chemists. This was not seen a year ago, and I can clearly see that there is a huge potential now. I remember 10 years ago, a lot of people were backing out of farm chem course because of various reasons. But now I can assure you that this course and this specialization is back with a bang. What is happening in our country is basically we are meeting 40% American generic drug demand, and we are supplying almost 25% total drug demand to UK. So it means we are funding these, I mean, we are supplying these drugs at a very cheap rate. So that's why they are buying from us. But somewhere we have to move ahead of this. I think we have not moved ahead because if we don't move ahead now, because this is the opportunity, it will really, you know, create a lot of imbalance in drug discovery market in the next coming years. So coming to vaccines, let me tell you, Serum Institute is world's largest vaccine manufacturer. Now he's manufacturing Covishield. So they meet almost 50% of their vaccine needs across the globe. I remember they have a, a, a pharma company even in Netherlands called Bilthoven Biologicals because they use that facility to manufacture vaccines for the European Union. So what is happening in all these aspects is we have a large pool of scientists available. So the problem in this is we are not using this talent as they have to be used. So now what happens in this scenario? Most of these people leave the country and go. So you will see a lot of scientists and abroad doing drug discovery aspects, because the opportunities for drug discovery in our country is very less. So that's why most of the scientists leave the country. It starts in 1940. I don't know if you know Dr. Yallapragada Subbarao, who was a LMS doctor from Visakhapatnam, went to United States in 1940 and discovered drugs. He was a doctor, but he was not a medicinal chemist, but discovered drugs like uh, tetracycline. He discovered drug like folic acid, methotrexate, diethyl carbamazepine. So these are drugs even today being used. He discovered, but nobody knows about it. But the thing is that we can do this. So the main problem that we see in this country is there is a lot of what you call uh, doubt in the industry academia interaction, which is very strong in the West, what I have seen. So the pharma companies usually invest a lot of drug discovery aspects in the academia, in the universities. And then they go, when they have something, then they go into the commercial aspects. So the main investments for this drug discovery comes into academia. So this is one we have to look at it. And India being a huge talent pool of scientific potential, I think the pharma companies and the industries have to take up this. I think there are some policies which have been placed now, so this will be improved in the coming time. So there is a talent pool of scientists. Even the biotechnologies availability is huge. So somewhere now the biologicals have taken a big leap. I hope this can lead to we can crack this drug discovery puzzle, which we are not good at it and we are lacking in it. And what happens in drug discovery? It's a long-term commitment. It's not like you will invest in 1 year, 2 years, you get results. It's a continuous process. Continuous process in the sense we have to start investing today. 10 years down the line, you have results. You can't have results till 10 years. So that is how the policies was made to the government this time, and they have agreed to some of these things. So the main aspects are we are only manufacturing API and bulk drugs. And with China, with the standoff that happened, political standoff that happened, so there has been a huge problem with supply of API and bulk. So basically, we are in a very bad shape of some of the APIs. And what happens? Cost of drugs will go up. So this has to be controlled. If we don't control it now, it will have a huge cost escalation on all the medicines that we consume. So what we should do, we should be able to manufacture these drugs in our country at a cheaper rate, but without compromising any quality. And the main problem has been is a distrust between industry and academia, because they don't trust the academicians are capable of doing any research and delivering them. This distrust has to be removed, because this is possible as long as government sits and brings things together. You'll be surprised after 2006, because I remember a lot of Chinese were sent abroad to get trained, and all of them have come back. And now what is happening here is 10% of the new drugs that have come into the market are discovered in China. This is an alarming thing to us, because there can be a huge threat, because if these drugs are not available to us, we will be in a deep trouble. So what happens here is a drug discovery, it has to be seen from different perspective. See, drug delivery is a thing which happens in a year or two, and the products are there in the market. But drug discovery doesn't happen. It takes time. 5 years, 6 years, 7 years, 10 years, and then you have a result. And when you have result, you have a huge what you call uh, output from these things. So government, let me tell you, because I do work with the policy making, so I am aware of certain things. Because the new science technology policies on the DST website, you can go through it. You can see there has been a huge impetus to put on pharma sector. Pharma sector will see a huge investment in the coming years, and it will not only from the government. I think private will also chip in because they have realized the huge potential. You remember the number of pharmacies that have come up in the post COVID era, you'll be shocked. Like in an area of 1 square kilometer, if there was 2 or 3 pharmacies, now we are seeing pharmacies increasing almost uh, to doubling almost. Like if there are 3, there are 6. If there are 6, they are going up to 10. So you can clearly see that people are seeing the potential of healthcare, and then you know how this is going to change the game in the coming years. Actually, the Department of Pharmaceuticals made a proposal a year ago that they will have a dedicated R&D head only to push new drug discovery and commercialization. And since this COVID stuck, and we need a lot of money for this, and the money that has been asked for this is 1,000 crores for the next 1,000 crores every year for the next 10 years. Pump this, if we will have an outcome soon. So, and one more thing is, our students have to take up challenges in innovation. If you don't take challenges in innovation, naturally you'll be left behind, because only an innovator takes things forward, and things will come out in fruition as a new drug or something, and which can take a huge change in the coming years. So I hope the Indian pharma sector will work towards this, and I think we will be able to manage this. And our Honorable Prime Minister coined this word of Atmanirbhar because we have to be self-sustaining in certain things. So what are the proposals that were made? One such proposal was made is, our government sends a lot of students abroad. They sent me 2 years. I got a Voice Course Fellow. I went 1 year to Netherlands. I got a ICMR DHR Foreign Fellow. I went 1 year. I came back. I learned a lot of things. I transfer this to my students, my colleagues who come for mentoring to me. So I help them out. But if we are standing faculty students in 10s, 20s, 30s, it's not going to work. We need students to be sent abroad in thousands, then the output will come. I don't know how many people will come back, but the people who will come back are going to make a huge change. But let me tell you, this is possible. If we increase the volumes and the quality, the quality of number of students, especially the PhD students, who have this program of getting trained in our country and going abroad, getting trained there, coming back, writing their PhD thesis, it makes a huge change in the way they're going to work and think innovation in a different way. It's not that we can't do it here, but the thing is we have less numbers. All our institutes which are in quality are in less numbers. So we need to build the number of quality institutes in hundreds. So if that is possible, and then things will take a different change. So for me also, for everybody, innovation is a everyday learning process. It is not that I have learned and I can move on. It is not going to happen. We need to do this at all, because I'll tell you why this Atmanirbhar thing has come into picture. Because a lot of money is spent on importing things. Because if a lot of things are spent, what will happen is you are going to spend your foreign exchange in buying these things, technology, chemicals, you know, uh, whatever we want to import from outside, even raw products if you want to. So this all costs money. So when this costs money, when your foreign exchange goes out, it's a currency thing that drives an economy. So what will happen? Your rupee will come down if a lot of foreign exchange is going out. So that's why we are in this situation, because we were dependent on a lot of imports. So now, if we become self-reliant and we become a strong export-oriented country, that's what China is now about. So they are going to mint a lot of. This imbalance will work very well for Indian economy, but it has to be a balance. You know, how much you import and how much you export. There has to be some balance. So what I'm saying is, this foreign exchange is one of the most important aspect which drives our country. Now, you know, we if we compare ourselves with China, China spends almost 54 billion, or sorry, around 30 billion for R&D of in research. We are spending hardly three, maybe 10% of them. It becomes a huge gap to match them. You take any area, it is a lot. You know why? Because their foreign exchange is they have exchange in more than trillions. So that's why they're able to control a lot of things. So what we need to do is, if we discover new drugs, we will be able to export and earn foreign exchange. That's the only purpose. If we import, the things becomes expensive here. That's the simple things. So what suggested is, invest 1,000 crores every 10 years for the next 10 years. 1,000. Bring pharma companies, bring academicians. We have a huge amount of drug related research academicians available. Bring them together, fund them, ask them for the output. It will always happen. And one more thing is, we should not be thinking of selling always cheap drugs. We should be thinking of quality drugs, quality of life. So this will make a huge change in these aspects. If we now, for example, if we decide this in the next 10 years, if we are going to discover 5 to 10 new drugs, so what will happen in the next 10 years? Like, these new drugs will bring in those 10,000 crores we invested. It will come in different times. It comes in terms of human resources, it comes in terms of what you call technology, it comes in terms of reducing the imports. So it has a huge economic effort actually. I mean it is going to affect economically to a country. That's how we need to build this. So lastly, this is the last side. You know, what are the few areas I would ask you to focus on in coming years? Because this is going to change the whole world scenario in the post-COVID era. So one is the usage of artificial intelligence in drug discovery has been humongous. So we use a lot of these things, and we should develop the usage of AI. A lot of pharma companies are using around in the developing lot of countries. We had one such conference in 2016 on artificial, but there was one speaker who came from Belgium, and he gave a real perspective how drug discovery is going to be. And this he gave us in 2016, and now we are already 5 years, and you know what more things are going to change. We need drugs for future. We don't know what are the diseases that they're going to be, and do we have drugs for them? And see, in the COVID situation, we were taken back food by at least for 5 years. Economy, everything was affected. Lives, jobs, everything affected in this post-COVID era. I mean, you know, this COVID-19 created such a huge imbalance actually. So we need this predictive modeling, and this is possible. A lot of people, you know, you have heard of this who did the computer generations, he did it in 1960s. What will be, and see how we have evolved in computers. It's possible even here. 10, when we had the Spanish flu 100 years ago, and we have one more pandemic after 100 years. See how things have changed. There were so many deaths before, and there was not many deaths here, but still a lot of things were affected. We don't know how climate change is going to affect our disease models. We need to really build models for how climate change is going to. I don't know if COVID-19 is an outcome of climate change. We have to really look at it, because uh, we if we don't do it now, it will be too late. We don't know what kind of diseases are going to come in future. And do we have a model how we are going to fight these new diseases? We don't know. We have to fight these new diseases, see that these diseases are taken care of. We have a, it's what you call most advanced testing models. For example, imagine we could bring vaccines to the market in 6 to 8 months. Tomorrow we may cut down these things to 3 to 4 months. It's possible. It's not because we never thought that vaccine would be out in few months, and they did it. And even our company in India could do it. So it means that if there is a goal and a target and the supplies required for that innovation, it is possible, because government funded Bharat Biotech and they came out with a vaccine in 6 months. That's possible. What are the future vaccinations we need? Covishield, which is one of the product of AstraZeneca with Oxford. This vaccine was being developed almost 6 years, since 6 years. Only when the COVID stuck, they started checking this vaccine. That's why they were the first who could cross the barrier of clinical trials and become the first vaccine. Of course, because they had some messed up some data, and finally it was Pfizer and Moderna who took the first lead. But the vaccine of the Oxford's AstraZeneca Covishield was being developed in Oxford University for the last 6 years, and that's all. Let me tell you, the demand for medicinal chemists is going to increase. It's going to give a huge market in the coming years. Let me tell you, 1 or 2 years, you will see how pharma companies will be hiring these people. And they know that now, because of the government's investment in this, going to 1,000 crores. So I am 100% sure that there will be huge demand for these medicinal chemists. So to keep up with these things, do we have a syllabus to meet these needs is a challenge you and I have to think. All academicians have to think, because are we ready for the post-COVID requirements of the pharmacy syllabus or the medicinal chemistry syllabus? So are we ready to focus this? Are we going to be futuristic in this? So this is something we have to look at it. The one who wins this race is going to be successful. The one who not is going to take a backseat. So this was something I wanted to speak and share with you about the drug discovery perspective in the post-COVID era. So yeah, have any questions, any doubts, please ask.
Thank you very much, sir, for enlightening us about the situation in the drug discovery post-COVID era, and what would be the policy changes which are required for the drug discovery in India. Now, I would like to ask participant, if you have any questions, you can unmute yourself and you can ask your questions. Alternatively, you can put your questions in the chat box. The session is open for questions.
I think the youngsters should ask questions. So I think there are no questions.
If there are no questions, we will close the first session. Our second session is going to start uh, as per the schedule. So we are going to take a break till our next session is, I think it's going to start at 12 o'clock. Let me just confirm it. Yes, our next session is going to start at 12 o'clock. We are done with the first session a bit early. So we will take a break till 12 o'clock. And because Dr. Vijay Thiruvenkatam will join at 12 o'clock sharp. So it is requested that all of you please join us at by 11:45 so that we can start the next session on time. Thank you very much, everyone, for joining us for the first session. And see you at the second session at 11:45.
Thank you, sir. Hi, Kapil. Are you there?
Hello, sir. Yeah.
Hi, Kapil.
Hi. Good,
Good afternoon.
good afternoon, sir.
Hi, hi.
I will
So,
just
yeah.
make you co-host so that you can share your presentation later on.
Okay, yeah,
Actually, I did not realize that you are there.
yeah. Okay, someone is, okay.
So we'll start in next 10 minutes because
Yeah,
I have
no
given
problem.
time 12 o'clock.
Sure. It is the same way, right? The share screen is
Yes,
the same.
yes, exactly.
Because this will stop another screen sharing. Do you want to continue?
I will stop my screen sharing
Okay,
after
okay.
giving brief introduction, and then you can start sharing your.
Sure. Okay. Yeah. So how are things, Kapil?
Going good, sir.
You are now in office, or
Yes. I have joined
uh,
office since
how
yesterday.
is Namita doing?
She is good.
And the kid,
He is also
baby?
good. He is also fine.
Good. Very nice. So is it streamed on YouTube also?
Yes. We are streaming it on the YouTube as well.
Oh, okay. Uh-huh. So I think you have to. I have to. You have to stop the PPT and then I have to
Yes,
turn
yes,
it on,
yes,
like
yes,
something
yes,
like
yes.
that. Okay. So you had classes today, Kapil,
Yeah, but I had to reschedule those because of the workshop.
and also next lecture is yours.
Yeah, at 2:30, I'm going to
2:30.
give demonstration.
Uh-huh. Okay. How is the situation, Kapil, there?
Sorry,
How is the situation there? The COVID situation.
it's it's okay. I mean, it's rising in some areas, but
It was some places were locked down, no?
not really. So they are doing it. I think only some societies they are doing lockdown, but not areas completely.
Okay. Uh-huh. Mm.
So we will start in 5 minutes more.
No problem. No problem. Yeah. So, Kapil, this is sharing is like one participant can share at a time. What
Yes,
is
yes, yes. So
that? Is
once
the one?
once I have given the introduction, I will stop sharing
Uh-huh.
from my side, and I think then you will be able to share your screen.
Okay. Okay. Uh-huh. Okay.
There are still few participants joining, so I will just wait for next 2 minutes.
No, no worries. No worries. Yeah.
We'll start.
Good morning, Dr. Vijay.
Hello, sir.
Hello.
How are you?
I'm fine. So, welcome to NMIMS.
Thank you.
Yeah. Yes, Kapil, you can take on.
Yeah. I think we will start now with our second session. Few people are still joining. So Dr. Ashish, if you see people are joining, can you please add all the participants so that I will take over
Yes,
the
yes,
session?
we'll do that. Yeah.
Okay. All right. Good afternoon, everyone, and thank you for joining the second session of the 2 days workshop. Uh, we have with us Dr. Vijay Thiruvenkatam. Before we start with the second session on the role of crystallography and its application in the drug discovery, let me introduce Dr. Vijay Thiruvenkatam. Dr. Vijay is working at Indian Institute of Technology, Gandhinagar since 2013 in the Department of Biological Engineering. His expertise includes structural biology and chemical crystallography. And he's working on the small molecule and macromolecule crystallography since past many years. Dr. Vijay, before joining IIT Gandhinagar, he has done postdoc at Harvard Medical School and Boston University, where he worked on crystallography of different important targets in different diseases. So his expertise is going to be helpful for us to understand the application of crystallography in drug discovery. He has more than 50 publications in reputed journals, and he has received several research grants from the government agencies, and he has also filed several patents from his work. uh, so Dr. Vijay is today he's going to talk on the chemical crystallography and its application in drug discovery. I welcome Dr. Vijay and I request him to take over the session. So I will stop sharing my screen, sir, and so that you can share your screen.
Sure, sure. Thank you so much, Kapil, Ashish, and yeah, everyone from NMIMS for giving me this opportunity here to talk about my research in this very important workshop on recent trends in drug discovery. Before starting my lecture, let me share the. Let me share. Yes. Kapil, are you able to see
Yes, yes, your screen is
the
visible.
slide?
Yes.
Let me put it in the slideshow. One second. Are you able to see the slideshow?
Yes, sir.
Okay, thank you. Thank you so much, Kapil, for a nice introduction about me and my research work. And yes, we have been closely working. I have to tell everyone this, that we have been closely working with Dr. Kapil uh, from Nasim Monji Institute. And so I basically come from the background of chemical crystallography. My research started with chemical crystallography, identifying molecules that are of pharmaceutical importance. And then I went on to do my research on biomolecular crystallography, where I have looked into the targets related to chronic diseases like Alzheimer's, cancers, and so on, where we do a lot of work on protein crystallization, structure determinations, and try to understand them using biophysical techniques, their interaction with drug molecules and the target proteins. So today I will be talking about which is very, very important in nowadays drug discovery research, and also it is part of one of the regulations in FDA, Food and Drug Administration, USA, where any drug, as you all know, any drug has to have or follow some of the rules and regulations to become a full administrative drug. So today, chemical crystallography stands in mid of everything in drug discovery, because chemical crystallography, as it says, it is dealing with chemical structures, let it be organic, inorganic, bioorganic, or metallo-organic compounds, which are mainly focused on drugs and as well as other applications in the industry, cosmetic industry, or any other industry. So crystallography as such is an very important research, where it gives you more information about how to tune a small molecule and make it as a drug. That is what is crystallography is all about. And getting to know about the 3D structures are one of the important regulations and applications in drug discovery. So uh, one second. Okay. So looking at chemical crystallography, this is from the book of Desi Raju. That is, I have given the references at the end. So this is called the crystal engineering. And Professor Desi Raju is one of the pioneering crystallographer, chemical crystallographer, and also he is top most crystallographer in crystal engineering, to engineer any small molecule to extract out its properties by tuning the uh, pattern of packing and as well as the molecular symmetry. So this is one such slide from his uh, book called Crystal Engineering, where molecule is paramount in chemistry. Yes, obviously it is extremely important for any chemist to have a very good molecule designed and synthesized. And when it makes an assembly of molecules, it has some properties associated with it. And like that, if you see a crystal, when it comes to a molecule, it turns into a crystalline form to make understand how the molecules are assembled or how the molecules are packed inside the crystal. That is what is crystallography is all about. And as we can see in this picture, as Casey Nicolau has rightly given the title, molecules that change the world. So any molecule, any organic molecule which has tremendous property as a drug, or which has tremendous property as toxins, are very important to understand and study the structural 3D conformations of that particular molecule. So it can change its properties if it changes the conformations. So what is crystals? So as a crystallographer, I would really look into a crystal. What is crystals? Crystals are nothing but a solid substance which has three-dimensional morphology. When I say three-dimensional morphology, it has particular shape, geometrical shape. It can be any geometrical shape that can take up in nature. It has to have a quality of something called the diffraction. I am sure that most of you know what is diffraction. It is from our textbooks of 10th standard, or even in our 12th standard. What is diffraction we have studied. Whenever light falls on some objects, it starts diffracting, it starts reflecting, or it starts scattering. So X-ray diffraction is one such phenomenon. When it falls on any crystalline form, it starts scattering. The phenomenon is nothing but scattering, and it is called as diffraction. And what is it scattering about? It is scattering the atoms, ions, molecules, amino acids, peptides, bigger molecules, protein molecules, DNA, nucleic acid. It is scattering everything that it comes in path of its when a molecule comes in path of its way. And what is it recognizing? It is recognizing how this can be extracted, how these molecular arrangements or atoms or ion arrangement can be extracted out from the crystal. Because just looking at a small tiny crystal under the microscope, you can't get any information until you pass on some high energy rays, that is nothing but X-rays. And it also has many other thermodynamical factors associated with it, like enthalpic factors, gain over entropy, where you have to have certain thermodynamical equilibrium inside the crystal that is maintained with the help of the electrons around the molecule, around the atoms. And this creates a thermodynamical entropy behavior inside the crystal. So crystal structure, as I told you, is nothing but the arrangement of atoms, molecules in a regular repeated manner, or very uniformly arranged in 3 dimensions. When I say 3 dimensions, it is in all the 3 directions of space, X, Y, and Z. And this happens in a form where the molecules arrange with the help of something called the intermolecular interactions, where the molecules within themselves interact in such a way that these interaction energies make it more stable to stay in particular conformation or in particular orientation in 3 dimensions. And this leads to the design of new solids or chemical properties of that particular molecule. Sorry. So the first X-ray crystallographic information of molecules was got from the Braggs. So both W.H. Bragg and W.L. Bragg, father and son, who started probing molecules, or you can say the first molecule that was visualized was salt, NaCl. And later on, they went on to explore more about the properties of molecules. Like here, what you see is a hexachlorobenzene and urotrophin, which is the earliest organic crystal structures that was determined. And it was determined to an accuracy that they could exactly tell the bonding between the two atoms and what is the bond length and how the electron densities are distributed. Given the molecular structure of a compound, what you can get is you can get the exact uh, arrangement of atoms inside the crystal. So then moving on to, we are not only looking at a single molecule, we are looking at an ensemble of molecules which are called the supramolecular entity. Why I am saying this is whenever there is a pharmaceutical drug, it is not only alone sitting with itself in a one particular space, but it is also having the neighboring molecules or atoms that are giving its stability, as well as it is helping in performing its properties inside the system. So this kind of arrangement or molecular ensemble using intramolecular hydrogen bonding, or you can call non-covalent interactions, is nothing but the supramolecular chemistry. When we call this as supramolecular chemistry is when we have few larger than 25, or if we have larger than 15 non-hydrogen atoms, which is forming an particular pattern in an given space, this becomes a supramolecular arrangement. And the molecule is also big. When I say a molecule is big, it is about non-hydrogen atoms, because hydrogen atoms are obviously included along with the atoms to satisfy the valency. So non-hydrogen atoms more than 15 becomes a supramolecular entity, or it becomes a supramolecular arrangement of molecules inside the crystal. So it becomes more complex, and it becomes a multi-component molecule which has many functional groups associated with one molecule, which is helping to form a larger ensemble of molecules inside the three-dimensional space. So here again, when we call it a supramolecular entity, it has to satisfy the periodic nature of molecules inside the crystal system. So what are the important parameters that are associated with this particular molecular arrangements? One is the equilibrium state of the molecule, that is the energy minimized state of the molecule, because that is the natural state of a molecule where all the parameters like vibrational, rotational, transitional parameters remain at the minimum energy, and it is not at the excited state or in the perturbed state, what is called free energy minimum. That is called the free energy minimum, and this results to the neighboring molecular arrangements, which is acting as an attractive and repulsive force. What are these attractive and repulsive force? These are nothing but the electrostatic interactions, the weak and strong hydrogen bonding, and different types of halogen interactions. When I say halogen interactions, it is atoms having higher atomic number, fluorine, chlorine, bromine, and chloro, and so on, which has higher atomic number, higher electron density, gives rise to something called a very defined halogen interactions. When I say defined, it is very directed, and it is very specific to that particular atom, which shows the property of halogen interactions. Apart from that halogen interactions, we also have the weak and strong hydrogen bonding interaction, and we also have the electrostatic interactions. These electrostatic interactions, which are called as ionic interactions, comes from salt, mainly in pharmacy industry or pharmaceutical industry. Whenever a drug doesn't have a high dissolution rate, or it doesn't dissolve at a very high rate, or it has very minimum energy to dissolve in a given solution, they tend to modify the molecule without changing its property by the addition of salts. So which is very important for a pharmaceutical drug, so that more the dissolution rate, more is the availability of the drug when it is taken or when it is administered. So these are some of the very important aspects of drug molecule in a pharmacy industry. So what drives all these things? So I am 100% sure that most of you would have opened your solid state book, and you would have seen this particular diagram. Without this diagram, I think nothing would have existed. So without this particular interactions or repulsions that exist between the molecules or the atoms, and without the minimum energy that is needed for the interaction and the attraction and the repulsion forces, we wouldn't have got a real good understanding about the molecules. So these are nothing but the intermolecular interactions. As you can see in this particular diagram, it is a potential energy diagram, and which is very important diagram to understand what is the energy that is needed for any type of interaction of a drug molecule or an API. Slowly I would get into the API concept, because API is also a drug molecule, except for the nature that not all the APIs can go to the final stage. Uh, Even though we have important APIs that are still yet to be explored and can improve the properties of those APIs. So these intermolecular interaction energies are keen or very important for any drug molecule. So one has to have an energy range of 4 to 80 kilojoules per mole. So how are these interactions divided into? In crystallography, particularly in crystal engineering, these properties, as you can see, you can take any molecule that you synthesize, you will definitely find these three general properties of interaction, which are called the strength, directionality, distance dependence. So the strength is nothing but the weak and strong hydrogen bonding interactions. The directionality is nothing but the isotropic and the anisotropic interactions. So isotropic and anisotropic, I would see it as a very crucial point at which the interactions are studied, because in an isotropic environment, we don't get more information about the other properties of atoms, but in an anisotropic environment, we start to get the atomic parameters, displacement parameters, what is called as ADPs, through which we can get back this information of strength of the weak and strong hydrogen bonding interactions, and also the distance dependency. When we say distance dependency, the distance between the donor and the acceptor and the angle between them should also be satisfied, or should also be present in your molecule to consider it to be a hydrogen bonding interaction or an electrostatic interaction. So as you can see here, these two molecules where you have the oxygen atom having the electron distribution around it. Oxygens are always the electron donor with the lone pair electrons on them, on the outer shell. So this gives them uh, the upper hand to interact with many of the neighboring atoms or the functional groups that are present in the molecules. Within the molecule, that is intra, or outside the molecule, that is intermolecular interactions. So here is a very nice picture which is taken from the same book of Desi Raju, Professor Desi Raju, where we all know that Gulliver story, Gulliver Travels, where the Gulliver was trapped by the soldiers that were who were tiny enough, but at the same time making a huge sense of tying him up as a whole team. Similarly, the hydrogen bondings or the electrostatic interactions are similar like this. It has a more strength when it comes together, and it makes the whole system under its control. So these interaction energies are very crucial for a molecule. And Then comes the van der Waals interactions, which are very important, and this is part of the atom energies, potential energy between the atoms, and the constants between them, and the charges associated with the two atoms. The first term is the dispersive or the attraction energy. The second term is the rise from the repulsive forces. That is, it can have two heavy atoms coming together very close to each other can have a repulsive force, and this would lead to several other interactions in the same molecule, other weak interactions. And third term is the empirical representation of electrical charge, electrostatic interaction. That is nothing but the charges associated with the atoms, if it is an ion, cationic or anionic atoms. So this is what you regularly read in your textbooks, but it is very, very important for a molecule, a drug molecule, to exist. Then comes the hydrogen bonding, the Pauline's principle. Linus Pauling in 1939 itself, he gave the thumb rule of how an hydrogen bonding should exist, what are the criteria for an hydrogen bonding, what are the distance that it has to have, what is the angle at which it has to, uh, if we have to consider it as a hydrogen bonding, what should be the angle. So these are some of the important aspects, and this is the thumb rule table for hydrogen bonding. You can consider this for every molecule that you synthesize in the solid state. Even in liquid state, you will find these hydrogen bonding interaction at a very high uh, thermodynamical equilibrium state. So, which is very crucial for any given molecule. So as you can see here, the table very clearly depicts these are the criteria for a very strong hydrogen bonding, stronger hydrogen bonding, and weak hydrogen bonding. So strongest, strong, and weakest. So that is how it is divided. Hydrogen bondings are divided, and these are the criteria it has to follow. Otherwise, it is not called a hydrogen bonding. Then comes the halogen bonding. There are several halogen bondings that has been discussed extensively in many research papers. The two recently, there are several halogen bondings that makes the property of a drug molecule to be more potent as a drug, and also there are properties of halogen bonding in heavy atoms or structural framework molecules, or metal organic framework molecules, where halogen bondings are very crucial for the stability of the whole system to exist. And as you can see here, we have CF4, CF3Cl, CF3Br, and CF3 iodine I. So all these molecules, all these heavy atoms, fluorine, chlorine, bromine, iodine, typically displays this type 1 and type 2 interactions. You can check it anywhere, you can check in any paper. If they have thoroughly studied the structural aspect of it, you will find one of these types. This is you can say this was extensively studied by Professor Desi Raju in his crystal engineering book, and it gives an very uh, elaborate explanation about what should be a type 1 interaction, what should be a type 2 interactions. Since time doesn't permit here, I just wanted to highlight, I would urge everyone to go and read this particular, you can buy this book. Actually, this is a fantastic book by Desi Raju. It tells each and every point about the uh, uh, interactions between the molecules in a crystal engineering aspect. And he was the one who first said that there are something called synthons, which is which you can see here. These are the hydrogen bonding interactions, but they are in particular form, or it is behaving in a particular manner, where you can see here the first OHO interaction, which is a typical dimeric interactions that you can see when there is a hydroxy group with another a donor group of O, which forms a homosyntons. It is called as homosyntons. Similar interactions that you can see, these are the patterns that regularly generated in many of the molecules. And there are something called the heterosyntons. These are the heterosyntons where we have a different functional group, NHN and NHO interactions forming a dimer, which is called the heterosynthon, and this is called the homosynthon. This is again called a homosyntons in chain, which is forming a network of interactions of NHO and the other side of NHO, where you can call this particular interaction as a bifurcated hydrogen bonding homosynthon interaction. So it is vastly explained and studied, and you can actually blindly follow these interaction patterns in most of your molecules that you synthesize. So there are CH pi interactions where these are also very weak interactions that can exist in a molecular packing. And this is kind of a heavy atom, iodine, interacting with the oxygen atom, which forms a typical tetrahedron geometry kind of interaction. And you can see Cl triple bond N, which is having a nice dimeric interactions. These are typical uh, motifs that you can observe in a interaction. So for doing all these studies of interactions, we need something called crystals. So what are crystals? Crystals are nothing but, again, the arrangement of atoms or molecules inside a particular direction in 3 dimensions. So crystallization starts from the nucleation and the crystal growth uh, technique, where crystallization can be done for most of your organic compounds, inorganic compounds, a complex of metal organic compounds, where you can find a nice arrangement of molecules inside a solid form. And all this is happening because of the thermodynamics equilibrium inside the crystalline form and the kinetic parameters of the crystallization. Kinetic parameters, when we say, it can be the temperature, it can be the pressure, it can be the type of solvent, it can be the type of pH that it is associated when getting crystallized, and so on. And based on all these parameters, they have developed a lot of models, simple models for crystal growth. That is given in this particular technique column, where you can see there are several other crystallization techniques. And these crystals can be of any geometrical morphology. So the techniques are solution based techniques, are nothing but the solvent evaporation that we usually do. So slow cooling, vapor diffusion, liquid diffusion. So in this solution crystallization, what happens is, it is the rate at which the solvent is evaporating. So, hello Kapil, am I on time or okay? Still,
Yes, you are.
yeah. So the solution crystallization, if you see, all of these methods depends on the rate of evaporation of the solvent. With the parameters of temperature, pressure, pH, and also the crystallization vessel. When I say crystallization vessel, the glass jar that we are going to use for crystallization makes a huge difference. So, sorry. And there are other methods that is called the hydrothermal or solvothermal crystallization, where you heat up the molecule and try to uh, cool it rapidly or very slowly to form the crystal, to form an arrangement, a periodic arrangement of molecules that eventually form as a single crystal or polycrystalline. And there are something called melt cooling methods, where you melt or you try to heat up the solid compound so that it goes to its melting temperature and slowly cools down. So in all these things, what is thermodynamically happening is the evaporation of the solvent that exists inside the compound or that is present in the solvent that is used for crystallization. Similarly, sublimation. Melt cooling and sublimations are more or less the same, where sublimation is you take a solid compound, final compound or intermediate compound, you heat it up to its melting point, because we can easily identify or calculate the melting point of a given compound by knowing its number of non-hydrogen atoms and the molecular weight of the compound, and reach up to the melting point and let it evaporate, or the moment it melts, it will start uh, melting at a very rapid rate, and it will start evaporating along with it. So the evaporation that condenses at another end of the vessel would eventually lead to very fine tiny crystals, which can, over a couple of hours, it can grow as a single crystal, which is very successful for a pharmaceutical compound. uh, if you have a melting point of a compound of 200 degrees centigrade, you melt the compound till that point in a closed environment. Both the ends should be closed, and it uh, evaporates and the condensation happens, and this condensation carries the molecules to form a crystal, which is very important for uh, pharmaceutical companies. So the regular uh, general solvent diffusion or vapor diffusion is what we usually do, and this type of crystallization is very successful. At the same time, it has to be fine tuned with respect to the solvent ratio or the physico-chemical ratio of the solute and the solvent. Without having that in the appropriate place, you may not end up getting a very good single crystal of the compound. So this ratio, weight by volume ratio, should be very important and at the very accurate at the same time. Then in all these four methods, so solvent, slow evaporation, vapor diffusion, slow cooling, liquid diffusion, in all these things it will work fine to get a very good crystal if you keep the physico-chemical properties of the compound and the solvent to be at a very critical ratio that has to be taken into consideration before doing the crystallization method. So other slowly moving on to the other crystalline solid forms of an API or a drug molecule are nothing but, as you all know, it is polymorphism, salts, co-crystals. The three important word in a drug industry is these three things: polymorphism, salts, and co-crystals. Because polymorphism salts co-crystals are also part of the FDA regulation to be satisfied for the drug molecule to go ahead for the animal studies. So we have to prove that any final molecule is either in a polymorphic state, does it behaves as a polymorph, it has a salt present in the molecule, or it coexists with another molecule. So these three important solid forms, not only forms, three properties, chemical, physico-chemical properties, are very, very important for an API. So these properties can differ among different polymorphism. Like you can see here, they have different color, compressibility, crystal shape, density, filterability. So all these different properties that exist with the molecule gives rise to the polymorphic form. And you can imagine how difficult is a molecule to come out as a drug until unless it is not, all these points are not ticked in their final compound. Otherwise it will not even go to the approval of FDA. So all these things has to be given, all these information has to be given, which is very, very critical, and this can be only got by crystallography. You can do NMR studies, but you will not see, because it is in the solution state, and all these are mostly in the solid state. All these are solid state properties. So other criterions for a drug molecule or an API is pKa of the active pharmaceutical ingredients, the formulation, which is called the previous clinical or market use, toxicology data, past use and success in screening studies of these particular APIs, ICH guidelines. So all these things are dependent on the polymorphic behavior of the molecule, and we do not know how it would behave when it is administered as a drug. So for which we initially screen all these things. Then comes the co-crystals, which is very important for a pharmaceutical drug. And many of the existing pharmaceutical drugs are named here, as you can see, and it is also administered, or it has also been taken as a tablet, tablet. So what makes a drug in all these things? Because tablets are solid state form, and when it dissolves inside a water, we have to see how it behaves, or how much it is available, or how much it is getting dissolved, for which we have a cofactor or a coformer which is associating it, or which is enhancing its drug properties, which is called as bioavailability. As you can see here, it is a very nice picture that is taken from this Nature review paper, where the bioavailability of a drug-like molecule is defined as the percentage of the drug which reach systematic circulation uh, inside our human body, or inside our gut as well as the liver. So how much it is getting absorbed during the process when it is taken as a tablet, that is what is called the bioavailability. In very simple terms, you can say like that. But for enhancing this bioavailability property, we need to have something called hydrates, solvates, co-crystals, salts that will enhance the property of the crystal. Because whenever we say we have a final compound as a drug, we have to test whether what is the dissolution rate, how much it dissolves in water, what is its solubility, what is its efficacy, or that will retain inside as a drug, or it gets metabolized and it goes excretes out. So at the same time, it has to perform its function as a drug. So all these things we have to study. So the role of co-crystals become very important. And as you can see here, carbamazepine, uh, what is it? Fluoxetine hydrochloride, antidepressant, antifungal, uh, and so on. Many such molecules, drug molecules, are having the existence of co-crystals along with it, or co-formers along with it, which is very important for a drug molecule. So as you can see here, uh, what is the important aspects as a co-crystal? What are the important properties that it has to have as a co-crystal? Co-crystals of carbazabine, uh, along with another co-former, sacrosine, can form very good hydrogen bonding. At the same time, it can enhance the property of carbazamine, which is relative, which can be seen as a um, in favorable of dissolution properties, suspension stabilities, and these are part of the uh, pharmaceutical screening of a drug molecule, and comparable oral absorption profile in dogs or any animal model that you can see as a drug molecule. So the 1:1 ratio of carbazamine and saccharine is very important to enhance the properties of that particular drug molecule, at the same time without changing the property of the molecule as a drug. And next is the co-crystals of triazole drug itraconazole, which is along with co-crystallized with dicarboxylic acids, which is very important in exhibiting dissolution profiles, because uh, itraconazole cannot alone can give a better property, for which they have added this dicarboxylic acid, which enhances the property as a drug, and which also helps in forming a better uh, solubility of the molecule and enhancing the bioavailability. More amount of compound or the drug is available when it is administered. So, and next is the tramadol hydrochloride, which is also a very important molecule in pharmaceutical industry, which is, so it is so important that it improves the pharmacokinetics behavior of the molecule by having this particular hydrogen bonding interaction with that particular HCl, what is called the hydrochloric acid, which is forming an NHCl interaction that enhances the properties of that particular molecule. So, and then comes the classification of a drug molecule, which is called the BCS classification, which is called the biopharmaceutical classification system. As you can see here, this is the typical diagram that you will see for many of your drug molecule, whether it is having class 1, class 2, class 3 and class 4. In which class does this particular drug molecule exists, whether it has high solubility, high permeability, low solubility or high permeability. So the class 1 is the one which you have to have for any drug molecule, which will enhance the properties of the molecule when it is administered as a drug. So, and here is an example of a drug molecule which is called the gefatinib, which is a very important drug molecule in cancer research and also administered as a chemo drug for several types of cancer. So what we did was we wanted to study gefatinib and see whether as we know from the literature, gefatinib exists in several different polymorphic forms. So, and it has the bioavailability is less. And we wanted to see what are the different polymorphic forms that gefatinib exists in and how to increase the bioavailability of that particular drug molecule, for which we started with a simple gefatinib molecule. We procured it from Sigma Aldrich and we started to explore more about gefatinib. And we know from the literature, gefatinib is a BCS class 2 drug. And we wanted to study its polymorphic behavior, and also we wanted to do a co-crystallization of gefatinib to enhance its bioavailability. So what we came across is, we know that gefatinib is an FDA approved oral administered anti-cancer drug, and it inhibits the EGFR of protein tyrosine kinase and suffers from poor aqueous solubility, that is 2.5 microgram per ml, which is very, very less as a drug molecule. No investigation of polymorphism, and there are certain forms but which doesn't have crystal structure from the Cambridge Structure Database, which is called the CSD. And the two forms that were reported, Thorat in 2014 highlighted two different forms of gefatinib. One form 1, which is the more stable form, and form 2 is a metastable form. So as you can see here, this is the typical form 1, how it changes form 1 to form 2 by the solvent, different solvents that we use for crystallization. And further, in the same paper, they further explored other forms of gefatinib using other solvates or other solvents that you can say, where they couldn't identify the crystal structure of the other forms, form 4, 5 and 6, but they could get to know that there are different other different forms as well with gefatinib. Form 3, form 4, 5 and form 6. And from here, we went on to start our study on jafatinib. And what we did is we started to crystallize jafatinib in the normal methodology of solvent evaporation with a stoichiometric ratio of the compound and the solvent. And we ended up by uh, looking into form 1 and form 2. We did get again form 1 and form 2, but we didn't proceed because it was already reported. And we started to look at the interaction patterns of these forms and what stabilizes the intermolecular interactions, because the molecule is huge. It can form a supramolecular assembly. And you can see here very nicely the motifs that we saw earlier in hydrogen bonding are appearing in the same way here, with the homodimer and heterodimer with the chain of interactions. Then while we crystallized the jafatinib, we got the molecule or the solvent molecule trapped inside the molecule, inside the jafatinib molecule. So as you can see here, we got crystals of jafatinib along with the solvent trapped as methanol. Methanol was the solvent that we used, and we got plate-like crystals, and we were wondering what would be this crystal morphology. And when we started to look into the 3D structure of jafatinib, we saw that jafatinib, the solvent molecule, one of the solvent molecule is trapped inside the jafatinib molecule. This is because of the hydrogen bonding interaction that is existing within the molecule, and also it is interacting with the solvent molecule. And we got to know that this particular crystal belongs to a different crystal system which was not earlier reported. It belongs to a triclinic system, and we solved the crystal structure and we said this as solvatomorph, because in polymorph there are different types of polymorph. One of them is solvatomorph, where solvent gets trapped while doing the crystallization. And we ended up solving the structure with the solvent, which was a new structure for jafatinib. And we explored the intermolecular interactions, and we saw that there is a solvent which is helping it to crystallize in this particular morphology. And then to further confirm that the solvent is existing in the molecule, we also did, even though we have the 3D structure, we wanted to cross verify with whether are we locating the solvent molecule as a proper entity inside the crystal structure or not. So we went on to do FTIR, where we saw the methanol stretching of OH stretching of methanol in the dimer, and we confirmed that it is rather the solvent that is trapped. Even though we solved this crystal structure and resolved the structure to 2.5 Armstrong resolution, or less than that, I think 2 Armstrong resolution, we could accurately determine the atomic positions. Then we compared our molecule, the methanol trapped jafatinib molecule, along with the already reported molecule, which you can see here, the conformational polymorph of green and red diagram, which is overlapped with the solvent trapped molecule. And we see that there is no much change in conformation, as you can see. Hello. So the red and the blue molecular conformation just overlaps exactly on top of each other, and we don't see that there is a change in conformation of form 2 or form 1, and there is a slight change in the angle of the phenyl ring that is associated, and um, along with the form 1, that is the green molecule. So but there is two molecules in the asymmetric unit that exist, and in the solvent morph, and we said that okay, this is the solvent morph, and it resembles the form 1, or it resembles the form 2, and we have the solvent morph of gefatinib. And we further went on to do some of the calculations of hydrogen bonding, where we identify what type of hydrogen bonding that enhances this particular solvent to interact with the gefatinib molecule. As you can see, there are several CH pi interaction and CHCl chlorine interaction, CHCl interactions, which are helping it to form the solvatomorph. Then we wanted to see if that is the case, if methanol gets trapped inside the molecule, can we try to have some of the coformers, which are called as generally allowed coformers in the FDA set of list, that can be used for any drug as a cofactor or coformer. You can call it as cofactor or coformer. So we went on to crystallize, we took some of the coformers from the FDA list, that is sorbic acid, cinnamic acid, we also folic acid we took, and several other coformers that we took for co-crystallizing it with gefatinib. So this led to co-crystallization of gefatinib with some of the coformers, which we solved the crystal structure. One such molecule is sorbic acid, which with the water molecule we co-crystallize successfully using the solvent evaporation method. And we studied the hydrogen bonding interaction in them, and we studied what is called very specific type of interactions that can exist, or that enhances this in coformer to exist together with the gefatinib molecule, that is called the Hirschfeld surface, or the 2D fingerprint and the 2D fingerprint plots, where it will tell you the exact contribution of OH, CH, NH, whatever type of hydrogen bonding interactions or halogen interactions that exist in the molecule along with the coformer can be studied very precisely. So we went on to study this, and we did the same study with sorbic acid. Sorbic acid with all the information put together here, and we did the same study with the cinnamic acid, where we were able to successfully co-crystallize cinnamic acid along with gefatinib. Then you will be wondering why, even after crystallizing all these things, how would the properties of the molecule enhance the properties of gefatinib molecule. So this is again the same thing. I will move on faster. So the critical point that we came into is to study these co-crystallized gefatinib molecule in cells. So we since our lab is also working on the protein biology or protein crystallography, we also do some of the studies in cells, human cells, or in bacterial cells. Since gefatinib is a cancer drug, we wanted to test in cell culture, human cell culture, or the mammalian cell culture. So what we did was whatever we co-crystallize the molecules, we took it along with only gefatinib, and other only cinnamic acid or sorbic acid or and DMSO, and we wanted to study the toxicity of the co-formers along with gefatinib. As you can see here, this is the typical cell, the preliminary experiment. Even though it is preliminary, it gives you an information about whether really the gefatinib co-crystal enhances the properties of gefatinib, or it gives you the same properties of gefatinib alone, or does it kill completely all the cells. So what we did was we did a cell viability experiment, where cell viability experiment tells you that how much drug uh, would take up by a cell in an environment, in a physiological environment, at what concentrations you can give this particular drug along with the co-former or along with the cofactor. So what we did was we took the cell viability, we did the cell viability experiment at different concentrations of co-formers and gefatinib, and only gefatinib. And as you can see here, the plot gives you this particular uh, um value gives only for gefatinib against what is called the HCT 116 particular cell line. This is a cancerous cell line which can be cultured in the lab, in the cell culture lab, and that can be used for any toxicity studies, cytotoxic studies against any drug-like molecule. So we did that, and we did take this HCT 116 cell line, and we at a particular concentration of this uh, drug, we saw that only gefatinib with sorbic acid, gefatinib with cinnamic acid doesn't have much increase in property, even though the addition of sorbic acid and cinnamic acid, it doesn't change any properties of gefatinib. Rather, it can exist along with gefatinib. So it doesn't enhances the property. Because we have to still do the solubility studies, which we are doing, and that will tell you how much it is soluble along with the coformer. At the time being, in the time being, we did this cell toxicity studies where it shows gefatinib along with the coformer doesn't change the properties of gefatinib. So this is one of the promising result, even though there is no change in the result, there is no killing of cells drastically with the help of coformers, but it says that it is more safer to have a coformer without changing its property. The property of gefatinib remains the same in both the cases. Rather, in sorbic acid, there is a slight uh, uh, improvement in the solubility or cytotoxicity of sorbic acid gefatinib co-crystal. And this further studies on solubility is still going on, and we would like to see whether the availability of gefatinib along with sorbic acid coformer is more, or only gefatinib is more. Does it increase the solubility? The coformer increase the solubility? That we have to check, and that we are doing. And we were successful in doing the co-crystallization of sorbic acid. As I told you, many other pharmaceutically allowed, generally allowed coformers, that are sorbic acid, mandelic acid, cinnamic acid, and risosinol, and several other allowed coformers are also being tested, and we could successfully do the structural elucidation. And one important thing that we noticed is the jafatinib with the coformer either forms a dimer or a monomer. In most of the cases, it is a monomer, and in some of the cases that helps in additional hydrogen bonding. As you can see here, the CH hydrogen CH interaction or the fluorine interaction gives rise to the dimeric formation. So these are some of the minute informations that you can get from a crystal structure that will tell us how to tune a coformer so that we get a more stable form, either a monomer, and it's improve its properties. So I would stop here. Uh, to end this lecture, I would say that there is several things that can be explored in crystallography, and it can improve the bioavailability of a molecule by studying the properties of the molecule in terms of polymorphism, co-crystals, salts. So since most of you are from the pharmaceutical background, I would really be interested you to see more work towards the physicochemical properties of the molecule by looking at these polymorphic behavior of the molecules, because it can drastically change the property of the molecule, and it can also stabilize the molecule as a, single entity in the physiological condition. So I would stop here, and all this work I have been doing with my graduate students, who all three of them have graduated out, and they are placed in different places as postdocs. And yeah, I am open for PhD students who are really interested in chemical crystallography and protein crystallography. Some of the students are still working on some of the aspects of jafatinib and also protein crystallography. And we have a combined group, or what we can call as a collaborative group, where all these people do a lot of crystallography work along with their own projects in cell biology or molecular biology. And I would really thank or acknowledge everyone who has funded for this particular work. First and foremost is IIT Gandhinagar, who has given us the infrastructure, lab facilities, and also the students of IIT Gandhinagar, PhD students, and also SERB for my first SERB core research grant on molecular biology of Alzheimer disease targets, and DRDO for jafatinib work, co-crystallization work of jafatinib, and also DST for giving us the first grant for developing or for having single crystal X-ray diffractometer in our institute. And also I would thank Harvard Medical School, where I still collaborate with my supervisor, postdoc supervisor for our molecular biology work, Dana-Farber Cancer Institute, where I still collaborate. These two places are one of the closest to my heart. And also one of my student also got Fulbright, and she also carried out some of the research work in Harvard Medical School and Dana-Farber. And some of the consultancy project which has also given us some of the drug-like molecules to screen for polymorphism, like Zydus. Zydus Research Center is one such institute, and Dr. Reddy's Institute of Life Science in Hyderabad is one such institute uh, who are happy to share some of the drug-like molecules to study for their polymorphism and co-crystallization. And the collaborators that have been, as I am telling, the Dana-Farber and Harvard Medical School. This is my postdoc boss, Jeffrey Wong, who still uh, appreciates my research work and also helps in collaboration with many protein crystallographers, and also Richard Bryce, who has been helping us in computational work of molecular modeling of proteins and small molecules. And also, last but not the least, Dr. Shivapriya Kribakaran, who is also a medicinal chemist and cell biologist, who with whom I take a lot of help in developing these setups of single crystal, as well as many more students from their group work in crystallography of small molecules. This is our setup of a single crystal X-ray machine, where we can do uh, data collection for small molecule drugs or small molecules, and also small peptides and small proteins that are of molecular weight of something like 10 to 15 kilodalton, that can be studied with using our facility at low temperatures and high temperature setups. And this is a small group that they who set up the whole single crystal facility, and most of them are not there anymore. They are abroad, or they have gone to other places to postdocs. And this is our fantastic campus, IIT Gandhinagar, which is exactly at the edge of Sabarmati River, and we have our research academic area where we have most of our research going in this particular area. And we are still expanding to other parts of the campus. I would be happy to take any questions. Thanks for being patient to listening to me. Thank you.
Thank you, sir, for such an excellent session. I'm sure that most of the pharmacy students or pharmacy faculty, they must have learned something new, because in pharmacy we do study medicinal chemistry and drug discovery, but our knowledge related to the X-ray crystallography or the small molecule crystallography is to some extent limited. So I'm sure that all of the participant, including us, must have gained some new knowledge regarding and how they can apply this knowledge for drug discovery. I would like to thank you for such a nice session, and
Thank you.
I would like to open this session for any questions from the participants.
Actually, I have to tell you, Kapil, this is only 5% of crystallography. There is a lot more to explore, because it is not only the structure elucidation, but you can fine tune your molecules to an extent that it improves its properties, pharmaceutical properties. And it is a vast area of research, but it can be done with the help of pharmaceutical research people, and as well as the basic science people, and also advanced science people. So
Right. So any questions from the participants? Dr. Ashish, you are on mute.
Yeah, am I audible now?
Yeah.
So I want to ask one question. Actually, many of the pharmacy people are dealing with the softwares, and they are trying to do some majority of the things virtually. So can you suggest few softwares where we can try to identify the proper crystalline form of the molecule, maybe small molecule or the polymer or the protein structure?
So the one that we use for identifying molecular properties is Cambridge Structure Database. Okay, you can make a note It is called as Cambridge Structure Database, which has a database of all the molecules in 3 dimensions. So that means you have the coordinates of each atom of any given reported molecule, and this database not only gives the 3D structure, but it also gives the information about the molecule, whether the molecule is has what properties those exhibits. Okay, and like it gives you the information about the molecule existing as a polymorphic form, or in what solvent it can be crystallized, or it can in what co-crystals that it exists with, several things, and their interaction, intermolecular interaction, several things can be studied. And also one most important thing is you can get the um, exact conformations of the molecule in 3 dimensions. So you get each and every parameter that can be related to your molecule, and see whether you can enhance your crystallizability of your molecule. So can we use different co-formers that can be used for crystallizing? Those information can also be got from this database, but this database is a licensed database, where I think it costs you 75,000 rupees, but it is in dollars, uh, something like $1,050 for institutions. So I think you can definitely go for this as a pharmaceutical, as a pharmaceutical institute. I think this database will be useful to understand the parameters of your for any given molecule. But this database doesn't carry any patented molecule. It would still be available, but if the patented molecule is reported in this particular database, that will be there. Otherwise, it will you will find the same patented molecule in SciFinder. So SciFinder is another database that can be very, very useful for your study.
Okay, thank you very much.
Any other questions from participants? I would like to ask one question from on behalf of all the participants, or from on behalf of our institute. You had shown the facility of the small molecule crystallography. So is it available for the outsiders?
Yes, very much. Yeah.
Okay. Thank you very much.
It is all the information is available on the website of IIT Gandhinagar. If you go to research facilities, we have put up a new website now, so you can find all the details about the charges, where to fill the form, and everything is online. You just have to fill the form and pay the charges, and we get the request directly, and we assign the particular slots for analysis. In the meantime, you can ship your crystals or compounds. Not only single crystal, we have all other facilities to determine the properties of molecules, like powder XRD, multi-purpose XRD, LCMS, and so on.
All right. Are there any questions before we end the session? So if there are no questions anymore, I would request all the participants to switch on their camera so that we can quickly take one snapshot with the speaker. So can you stop sharing your? Yeah. Thanks a lot. It is requested that all the participants, can you please start your camera for quick photograph?
बोलता है मी नाही का वेळ तुम्ही केलं होतं का रजिस्ट्रेशन काय होतं तू माझ्या याच्यावर क्लिक केलं असेल नाही हे अगं मी अन्य
All right, thank you everyone. Thank you for attending this session. We are going to close this session. The next session is going to be at 2:30 p.m. So there would be a break of around 1 hour, and that would be a demonstration on some of the chemistry tools. Some of the tools are very basic because we have a lot of student participants. So some tools are basic, but there is one very interesting tool which can be of interest for almost all participants, which can be used for the optimization of molecules for drug discovery. So please join us back at 2:30 p.m. So see you again later. Thank you. Thank you.
Thanks,
Thank you,
Kapil.
Vijay sir. Thank you,
Thank
Vijay
you.
sir.
Thanks, Kapil. Thanks, Ashish. Thanks, everyone. Thank you.
Thank you, sir.
Bye bye, sir.
Thank you. Bye.
So good afternoon, everyone. Welcome back to this post-lunch session where we'll be having hands-on different chemistry softwares. We'll learn how to draw structures and how to calculate the physico-chemical properties and how to convert the structures into different formats. So that will be helping us in tomorrow's session. So we'll wait for another 5 minutes. We'll wait for other participants to join, and within 5 minutes or in next 5 minutes, we'll start with the session. Okay, so we'll start now. In this post-lunch session, we have Dr. Kapil Julie with us. He's assistant professor working with SPPSPTM and NMIMS University. Dr. Kapil has completed his PhD from the University of Bonn. He has completed his PhD with specialization in medicinal chemistry, and after that he has almost 3 years of postdoc experience, 1 year in University of Bonn and 2 years of postdoc experience at IIT Gandhinagar. And after that, Dr. Kapil is associated with us. Dr. Kapil has one patent which is granted, and one patent is under process. Dr. Kapil also has more than 27 international publications and is having more than 4 grants, which includes certain government grants and industrial grants. His expertise is in cancer, then drug discovery and organic chemistry. So with this brief introduction, I request Dr. Kapil to start with his session. So over to you.
Thank you, Dr. Ashish, for introduction, and welcome all of you again for the third session, which is a demonstration on some of the tools which can be used for organic chemistry, but also which are needed for the many of the drug discovery processes. Now, when we talk about different softwares for chemistry, uh, and their application in drug discovery, they are not necessarily needed for some of the theoretical calculations which are done for drug discovery, but also you need such softwares for making reports, making publications. So I'm going to start with two of the very common softwares which are used to draw structure and manipulate those structures and to make reports. One is ChemDraw and second is ChemAxon. I'm pretty sure the faculty members who would be here, they must have used these softwares. So initially I will go quickly through these two softwares. So because there are some of the students are also there, and I'm pretty sure that these softwares would be useful for them in their further studies, as well as if they are going to enter in the research area. And the next part of my talk or the demonstration would be on the Data Warrior. Now, Data Warrior is an amazing free software which can do so many things. And the demonstration which I'm going to do is the application of the Data Warrior in optimization of the molecules from the different sources. If you have molecules and if you want to do the lead optimization, you can very nicely use the Data Warrior to visualize different parameters, different calculations can be done using Data Warrior. So we'll discuss over it further in detail. So let me share my first presentation. So what I'm going to do first is that I'm going to uh, show some, I have prepared a few slides about the softwares which I'm going to talk about. And in the later part, I will show the demonstration. So I will not take too much time explaining about this software. Maybe like 10 minutes, next 10 minutes, I will talk about it. And then we will move straight away to the demonstration. So as I said, I'm going to talk about ChemDraw, ChemAxon, Data Warrior. And as Dr. Ashish said, I'm also going to show the application of the OpenBabel for converting the formats of the structures from one format to another, which is very much important when you are using different softwares in drug discovery, especially for the theoretical calculations when you are doing in silico studies. For example, molecular docking, which Dr. Ashish is going to show you in the next demonstration tomorrow. So let me just quickly go through the first two tools, or rather there are many tools are available for drawing chemical structures. So chemists generally represent structures in their own preferred form. Like if I'm teaching to my students, I'm going to draw the structures on the whiteboard. If students are writing those structures, they are going to write on the paper. And obviously, if I want to publish my results or my research work, I'm going to put those structures in publications. Now, when I think about those drawing structures, every chemist or every person will have their preference or the preferred style of or the representation of the structure. Now, it creates a problem because the structures would not be uniform. Especially when I talk about the publication or when you are making report, it is essential that your structures are uniform. And here is where the molecule editor programs or the structure drawing softwares would be very much helpful. There are several such programs are available. Some of them are paid, some of them are free. I'm going to pick two of those which are the most commonly used and which I prefer also for drawing structures for making reports or for any other work. The first one is CameDraw, which is from Perkin Elmer. I'm going to briefly give you the history of the CameDraw as well. But the problem with the CameDraw is it's a paid software and it's very, very expensive. So the very good alternative to the CameDraw one can use is Marvin Sketch, which is from CameAxon, and it is free for academic use. So you just have to register on their website and you can download it for free and you can use it. There are other tools also like CameSketch, ACD. I find one problem with CameSketch is that it has not changed its interface since many years, although there were many updates. And hence I find that the CameDraw and Marvin Sketch are much more intuitive or they're much clear. Their interfaces are very clear, easy to use, and that's why I'm going to talk more about the CameDraw and the Marvin Sketch. And later on, I will show the demonstration of Marvin Sketch, which is a free tool. Now, when I talk about CameDraw, it was first developed long back in 1985 by David Evans and Stuart Rubinstein. These were Harvard University PhD students. When they were doing PhD, what they realized that they want to draw structure, but there was no software available for drawing structure. So they were using programs just like a MS Paint. Obviously, they were using Macintosh that time, but they were using programs very similar to the MS Paint. The problem with the MS Paint when you are using lines and drawing structure is that it doesn't understand the language of chemistry. What I mean is that you cannot do any manipulation, you cannot calculate any properties, you cannot have uniform bond lengths. So there were many problems when you use imaging software for drawing structures, and that is why these PhD students they designed this new software. So in 1985 they developed this CameDraw software, which was in earlier 10 years it was available only for the Macintosh, and since 1993 or 1994 the software was made available for Windows as well. In the later phases, they also introduced the BioDraw. So BioDraw it comes as a package with the Chem BioDraw tool, and the BioDraw has amazing templates which can be used for drawing biology. So those who are working in medicinal chemistry or even in the molecular biology or any biology fields, this software is amazing. The only drawback, as I said, is that it's very expensive and it's a paid software. So this software is designed for scientists, students, or scientific authors. It's a very powerful but very easy tool, and that's why I really like the CameDraw. If anyone wants to try this tool, you can go to the website of CameDraw. You can just search CameDraw from Perkin Elmer, and you can download 30 days trial of this software, and you will those who have not used this tool before, and you will love it. Similarly, CameAxon, it was founded quite later on in 1998, again by two brothers, Ferenc and Peter. The first CameAxon software product was Marvin, which is a chemical editor, which is still available. And it was first launched in 1999 for public. In 2004, CameAxon decided to support academic research. So they made all the packages which were available from CameAxon for free for academic use. But as of now, if you go to the CameAxon website, you will find that not all packages are free. Now they have made only some of the packages like Marvin Sketch, which was earlier known as Marvin Beans. They have made it free. So which is good enough for our purpose. That Marvin Sketch, it has so many things which one can do, well, not only drawing, but one can do many things with those. So where do we use this CameDraw and CameAxon, the Marvin Sketch? These are obviously used to draw simple two-dimensional represent of the organic molecules. But there are many other things one can do using these softwares. Like for example, if you want to generate a chemical structure from the name. Now where this would be useful, let's say if you have a drug molecule or a macromolecule which has a very huge complex structure, drawing such structures would be very difficult. But with these softwares, you can simply convert the name, whatever name if you have, into the structure, and vice versa. You can also convert the structure into IUPAC name. Now, obviously for students, it would be very interesting to check their understanding of the IUPAC naming, because by converting structure to name, they can cross check if the IUPAC name they have thought about is correct. Also with this software, you have direct access to analytical and chemical properties. I'm going to show you how you can calculate different chemical properties from the structure and fragments of these molecules. There are so many shortcuts available, so it becomes very quick. You don't have to really use mouse all the time. There are very nice shortcuts available which can be used for not only for drawing but for scaling and reporting. There are many pre-drawn graphical templates available for chemistry, chemical biology, and biology, but this is only available in ChemDraw. In ChemAxon, it is not available in the free version of Marvin Sketch, but in ChemDraw, which is a paid software, you will have all these tools available. Now I'm just going to show you before we move to the demonstration, what is the interface of these softwares and why I liked it. If you look at the interface, it's very clean. On the left-hand side, you have some tool to draw some bonds, to draw multiple bonds, to show wedge bond, or to draw arrows, add charges. And apart from that, you have many more templates, like you can put TLC if you want to show the thin layer chromatography in your report, you can do that. You also have the main tool. There are rings available which can be used to draw different aromatic or cycloalkyl rings directly. So it's a very clean interface and very easy to use.
There is some technical problem. I'll sort it out in 1 minute and we'll start it again. So please be connected. We'll start it again within 1 minute.
I'm sorry, everyone. I lost my connect. I will continue. So can anyone just please confirm if I'm audible?
Yes, you are audible. Okay, so with the use of these softwares, what we are going to do tomorrow is we'll understand certain properties of drug molecule, and that will help us to understand how drug are interacting with the protein structure. Okay, so Dr. Kapil is here, so we'll continue.
Yeah, so sorry everyone, there was some internet issue on my laptop. There was some Wi-Fi issue, so I hope I'm audible now, and my screen visible to
Yes,
all of you?
yes, yes, yes.
Okay. All right. So what I was talking about was the interface, and as you can see, it's very clean interface. And here you can see this is the interface of ChemSketch, which is very similar to that of the ChemDraw, where you have different tools available, like you have the bond drawing tools, or you have the templates, like you have the different rings, aromatic rings, and on the right hand side you have different atoms which can be easily introduced into your molecule. So these toolbars actually make it very easier to draw structures. So this is just a comparison of these toolbars available in the CameDraw and CameMaxon. I will just keep it. And this is just in advance. I mean, for the arrows also, as you can see here, you have different types of arrows depending on if you are showing reactions or if you are showing mechanism. You can use curve arrows, or if you are showing equilibrium, you can use the other types of arrows, the equilibrium arrows. And as I said before, there are so many templates available in CameDraw. Now, these kind of templates are not available in the Marvin Sketch, but CameDraw, if you get access to it as a trial version, you can always try those. You have very nice templates for drawing different diagrams. You can show your reactions, you can reaction setups, and you can do so many things. Now in Marvin Sketch also you have templates, but those are restricted only to the aromatic rings or bicyclic rings or the molecules, but not, you don't have other types of templates like you have in the CameDraw. Now, one more thing I wanted to talk about is the advantage of using these softwares is you can convert one style to another. Now, if you look at the different structures which I have put here, I mean, it's only one same structure, but it is in different style. What I mean by style, you can see that the bond length, bond width, bond angles, they are slightly different when you go from one structure to another. I can compare, for example, American Chemical Society, ACS, structure, and Journal of Heterocyclic Chemistry. You can see both of them use different font. ACS uses, I think it's Arial, and while in the Journal of Heterocyclic Chemistry, it uses Times New Roman. So you can see there is a difference. In RSC also, you can see the bond width, the thickness of the bonds is different as compared to in the ACS. So with these softwares, you can convert from one form to another, and that is one of the advantage of using this software. By one click, you can have the required style of your structure, which is necessary when you are publishing your data. Now, this is just an example where the ACS publication, they give specifications like what should be the chain angle, bond spacing, what should the length, what should be bond width, line width, etc. What should be the font? Everything is given here. Now, if you have to do this manually, it would be really cumbersome, and that's where these softwares are very useful, because you can just go to the setting, change the setting of the style, and that's it. You will be drawing your structures with these specified settings without having to change each and every parameter. So this is just how you convert from one form to another. This is in the CameDraw. You can see that there is something called as applied document setting, and you can see there are so many templates available like ACS. You have the Journal of Chinese Chemical Society, Journal of Heterocyclic Chemistry, for example. So you can just select those templates and you can convert your structures very easily. But if you don't apply this template and you have already drawn your structure, that then also you can change the template. Oh, sorry, change the style. You just have to select your structure, go to the object setting, and you can see here so many options are available for changing the setting or the changing the style of your given structure. Now, the next one, which is important, is the structure cleanup, which is possible with the help of these softwares. And this is very important. Now you can see that the structures which I have drawn purposely, obviously these structures would not get drawn like this, but I have drawn those purposely, and you can see the bond angles, bond lengths, they are not really uniform, and the structure doesn't look really good. So with these softwares, and when you're drawing big molecules, macromolecules, sometimes it happens that the structures are not really looking good, or there are some overlaps of the bond. In such case, you can use a simple command, which is a cleanup structure, which regularizes the bond angles as well as bond lengths. So you can see that the structure on the left-hand side, how it is looking now, all the bond lengths and bond angles are uniform, and it is looking much better. Similar is the case for the down one. I think there is one more example here. You can see that this is a cyclic compound. And now this, I have not drawn it purposely, but when you draw this structure, it comes out like this, which is not looking really good. So you can just select the structure, go to the structure menu, and use the cleanup structure, and your structure would be in the much better form where the bond lengths and bond angles are uniform. The next advantage of using this tool is annotation. If you are publishing your work, you can, and you have to do some annotation, it can be very easily done. And you can use different colors, different arrows, and then it can be used in your reports. The next thing, which I told you, the advantage of these softwares is the converting name to structure and structure to name. This is an example from CameDraw. Now you can see here there is a one large molecule which is amphotericin. Now no one will remember this structure. You can see there is a lot of stereochemistry also involved. And if you are trying to draw it manually, it may happen that the structure will not look good, as well as there may be some errors when you are drawing, you are not taking care. But because these softwares, they have so many compounds already in their databases, you can simply search, you can simply convert either the IUPAC nomenclature into the structure, or you can simply use the common names. Generally for many of the drug molecules, the names are already stored in the databases, so you can simply convert the name into the structure. So you can use a common name, or you can use also the full chemical name to convert them into the structure. So this is one more advantage of using these softwares. Similarly, you can convert structure to name. As I told you, especially for students, this would be very nice tool to confirm if the IUPAC nomenclature they have written is correct. So you can see here, I have a structure of caffeine, and I wanted to convert it into the IUPAC nomenclature. So you simply have to select the structure, go to the structure menu, and you can simply click on convert structure to name, and you have the name of that compound. Now, the next important thing, calculation of chemical constant. Now, this can be done with the help of both CameDraw as well as CameAxon you can use. Now, whenever you are doing drug discovery, or you're doing a synthetic chemistry, you need to calculate molecular weight. When you want to confirm the mass of the compound, you need to calculate the exact mass. Or you may want to calculate chemical formula. It can be done very easily using the analysis. So you have to again select the structure, go to the structure menu, and you have the option to, or you can simply right click and you have the option to generate all these properties. Another way to have a look at these analytical constant is to have this analysis window always open. Now this is available in the CameDraw, not in the CameAxon, Marvin Sketch. So here you can see that if you have this window open, and when you draw any molecule, you will see all the properties of these molecules, like molecular formula, exact mass, molecular weight, as well as you will see the fragmentation pattern which is possible for these compounds, as well as elemental analysis. So this is very nice tool to have. One more additional important thing which is available in CameDraw is NMR prediction. Now sometimes it becomes difficult, those who are entering newly into the NMR chemistry, or they want to predict the NMR, they want to confirm the NMR of their compound. They have taken the NMR sample, and they have got the spectrum, NMR spectrum, and they would just want to confirm if it was right or wrong. They can use this tool to predict. Obviously, the prediction is not always correct, but at least it gives you some idea about the chemical shifts of different hydrogens. Now this is 1H NMR, so it will show the chemical shifts for the different hydrogens which are present on the molecule. So this is one good addition which is available in the CameDraw. So before I go to the demonstration, just let's have a look at what are the advantages of using CameDraw and Marvin Sketch. So the first advantage is that it is very easy to draw structures. You can easily convert name to structure and vice versa. You can do cloning of molecules. That means you don't have to draw the same structure again and again. Now this is really useful when you are drawing a reaction. During the reaction, you must be showing some small transformation in the molecule, so you don't have to draw that molecule again. You can clone it with a click of a button, and it saves a lot of time. You can also check the structure if you have drawn the structure which has the valencies and the bond length, bond angles, if they are correct or not. You can do that. You can clean the structure as we have seen. Also, it helps us to copy the structure elsewhere. And once you have copied the structures in, let's say, in the Word document or in the PowerPoint, you can just double click it and you can edit those structures again. So that is very much advantageous, that the editability of these structures are retained when you copy those structures in any of the document. So there is availability of lots of shortcuts and templates, especially in the CameDraw, and BioDraw is amazing. But again, as I said, BioDraw is not freely available. CameDraw is not freely available. So for CameDraw, the paid version costs around $584 US dollars per year, which is very expensive. Or if you want a perpetual license, it costs around $2,000. Now, for students, maybe the cost is slightly lesser, but still it is very expensive, and it makes difficult for us to buy until unless the institute or university buys buy those licenses for the CameDraw. But there is nothing to worry because we have the Marvin Sketch, which does almost all of the important job except the templates, which are not available in the Marvin Sketch. But for drawing and calculating different properties, Marvin Sketch is very good software, which is free for educational use. You just have to do the registration. So now I'm going to move to the demonstration. So let me stop sharing my presentation and share my desktop. All right. So this is Marvin Sketch. I'm going to first start with Marvin Sketch. Now, as I told you, you can look at the interface of the Marvin Sketch. You have a very clean window available in between where you can draw structures. On the left-hand side, you have some of the templates, like you have some heterocyclic ring, you have aromatic ring, you have the cycloalkanes available also, and you have the bond tools. Now, when you want to draw multiple bonds, you can just click on that arrow and you can see that you have option to draw double bond, triple bond, or wedge bond, or the dash bonds. You can do all those things, or you can show the double cis or trans bond. Many things can be done. Also, if you want to draw a chain, you have a tool available. Then if you want to add some text, you can do that with the text tool. You have arrows like single arrow, retrosynthetic arrow, equilibrium arrow, resonance arrow, curved arrows. So you can use these arrows to write your reaction, write the mechanisms. Many things can be done. And there are some brackets available which can be used, or the shapes available which can be used for the annotation, or for if you want to make the reports. And here are the charges. If you want to put some format charges like positive charge or negative charge, you can use these tools. And on the right hand side, if you look at, you have some very commonly used elements available, although we don't need to use those always, but they are handy. And you also have the periodic table available. So when you click on this small icon, you will see that it opens a periodic table which can be easily used for drawing structure. So let us try to have example. If I want to draw a structure, I'm just going to click on the bond icon. So it brings one stick, as you can see, which indicates actually CH3, ethane. So if I just click there, let me zoom it so that it is visible to all of you. I'm just going to zoom a bit. So I hope it is visible clearly. So when I click anywhere, you can see that it adds CH3, which is ethane. So let's say if I want to draw propane, I'm just going to go to the one bond, and it shows a green dot, which indicates the bond would be added there. So if I click here now, it adds one more bond. Now I have propane. Let's say I want to add double bond. How will I do is I will go to the middle of the bond. You can see it shows that a green colored bond, which indicates that if I click there, it's going to add additional bond. So it's very simple to draw structure. Now let's say I want a triple bond. I am going to click it again. I want single bond. I'm going to click it again. So very easily I can do that. Let's say I want to draw ethanol. One option is I can go to the periodic the periodic table, which is available on the right hand side. Select O, come here. I have that green dot. I click here. I get the ethanol structure. The other alternative, I'm just removing it. I'm going back to the stick. Another alternative, you bring your cursor to the carbon where you want to replace it with a hydroxy group or the alcohol. I click O on my keyboard. I'm not using the oxygen there. I'm just clicking O on my keyboard. You can see that I can very easily get the structure, I mean the ethanol. Let's say I want to draw ether. What I have to do is I just have to click on go to the oxygen. You will see the green dot. And I click it there, it adds one more carbon now. Now it's a ethyl methyl ether. So very easy to draw these structures. Let me go to the cyclic compounds. Now, if I want to draw cyclic compounds, I'm going to use one of these templates which are present on the left hand side. Let's say I want to draw cyclopentane. I can click it here. I just put it here. It draws a cyclopentane. Let's say I want to add oxygen here or nitrogen, for example. I've done oxygen already. I'm going to bring that my cursor there. There is a green dot, and I'm just going to click N on my keyboard. So I don't even have to select the atoms from the right hand side bar. You can do that, but if you just click N, it is going to save my time. So let's say I make it heterocyclic compound. Let's say I want to draw indole. I'm going to take the benzene ring now, bring it closer where I want to have this benzene ring now. You can see that you will see a pink colored dots. You just have to click there. Now I have indole. Very easy to draw. Let's say I want to add some substitution on the nitrogen. Again, I'm going to select that line, bring my cursor there, click there. So now I have N-methyl indole. Let's say I want to add ketone now. So it's N-acetyl indole. You can see that very simply. You can draw this. Let's say I want to add phenyl ring at the third position. I'm just going to click it there. I will go to the benzene ring on the left hand side. Come here, you will have that green dot, and I simply draw. Now you can see that there is some overlap between two molecules. You can simply select it and oh, that has distorted. I can select it and move it away. Now, let me show you generation of the name of this structure. So what you have to do is, now I have two structures here. If I go for directly generating structure without selecting, it will generate a name for both the molecules. I want to generate structure name only for this. For selection of any structure which you have drawn, you have a marquee tool here. You can see up here, you have a rectangle selection, you have a lasso selection. So you just click on that. You can select your structure, and once the structure is selected, you go to the structure menu, and there you have a structure to name. So you just click on that. You get options. Do you want a generic name or the traditional name, or you want to have the preferred IUPAC name? So I'm going to click on the generate preferred IUPAC name. I click OK. You can see that there is a generation of a box which gives us the name, the IUPAC nomenclature of this compound. So I hope you have understood. You can use this tool to generate names. Let's try to have have one example where I can convert name to structure. So again, I have to go to the structure. I will go to the name to structure. So let me change it to something else now. I'm not sure if in the KMAX zone Marvin sketch, if the drug names are available. Let me try it. I'm pretty sure that such names can be converted into Chem draw. So let me take example of gefitinib, which was used by Dr. Vijay Venkatam in his talk. I click on import, and you can see that, okay, it works. So you can see clearly that I didn't have to write this name, sorry, the structure of this compound. I just I just went to the structure, to name, and just I wrote gefitinib, and it has drawn the structure of gefitinib. Let me give you one more example. So instead of gefitinib, let us take then imatinib, which is also another tyrosine kinase inhibitor. So I'm just writing imatinib, and I'm going to click now on the import. So you can see that it's a quite huge structure, not very huge, but still it has a lot of aromatic rings, a lot of bonds are there, and it saves me a lot of time using this software because I'm just writing the name of the compound or the drug molecule, and I'm just converting it to structure. Another way you can write names of this structure is by using if you have the smiles available. Now, let me go to the PubChem. I hope my browser window is visible to all of you. So I'm going to write PubChem. PubChem is a database of many chemical structures. So let's say I want to draw the structure of amphotericin. We have already seen that it's a huge structure. So let me open it. So I'm going to search for amphotericin. Here is the amphotericin. I will open it. And what I'm going to do is I'm going to copy smiles. Now, smiles are another way of representing structures apart from IUPAC nomenclature. So you can see you have the canonical smiles present here. I can simply copy these smiles from here. Now, there are many databases available for the compounds which are present in the form of smiles, and those are really useful because you can, I mean, these softwares are very useful because you can simply convert your structures into, I mean, the smiles into structure. So what I have to do is I have to go to the edit and I just click on paste. You will see that I had copied the structure as smile. I didn't have to do anything. I've just pasted the structure here, which was copied as a smile, and you can see that I've got the structure with the correct stereochemistry with all the bond order and everything is correct. So I hope you have understood how I can use this software to easily draw structures, convert structure into name, convert name into structure, or convert smiles into structures. Now what we will do is I will go back to the Graffiti nib again. So let me go to the structure, name to structure. I'm going to write Graffiti nib, import it. And let's try to calculate some properties. As I told you, for the drug discovery, many of the in silico studies, you need to have properties, different properties of the molecule. Let's say a simplest property one can calculate is the log p value or the lipophilicity, which is very important for the pharmacological activity or even for crossing the drug molecules across the cell membrane. So when you want to compare different drug molecules and their activities, in many cases the log p is one of the criteria which is considered. So again, if I want to calculate log p, I'm going to select the structure. I'm going to go into calculations. Now here you can see there is a window called calculation. You can see you can do so many things. You can do the element analysis, you can calculate the pK values. I want to calculate now log p, so you can do the go to partitioning and calculate log p. Even you can determine this aqueous solubility. Here also you can predict the NMR. You can predict C13 NMR and 1H NMR. I think you need license. Let me try it later on, but I think you need an extra license to be purchased for that. You can generate different conformers. You can generate topology. You can do the topology analysis. You can calculate the polar surface area, which is also one of the criteria when you are talking about the activity, or when you are comparing the activity, which is one of the parameter when you are doing the QSAR analysis. So let me try to show the log p value. I'm going to go to the partitioning, click the log p, and it asks there are different methods. So I'm just going to use the first method. You can use also KMexon. You can have a look at it, how it uh, differs. And then I'm just going to click on OK. It opens a window and says that I don't have a license. As I said, for the calculation, we don't have license, but again here, nothing to worry, because we get the results, we cannot save those. But at least when I want to do simple calculation, I can freely use it. You get a warning that there is no license available. But if I click OK, I still get the result which cannot be saved. But at least you can generate. So you can see that for this molecule, the log P is calculated as 3.75. So I just wanted to show you one example how you can generate different properties, or you can do different calculations with the help of the free software, which is Marvin Sketch. So this is about generating properties. I'm going slightly quickly. If you have any questions, you are free to stop me if you don't understand, or if, especially for the students, if you have any doubt, you can stop me and ask me anytime. Now, next thing, which is important for tomorrow's demonstration, is saving these structures. Now, for the molecular docking, which will be taken by Dr. Ashish tomorrow, you need to save these structures in specific formats. Now, when I save this structure, I just simply have to go to the file and go to the save. Let me go to the desktop and create one new folder for demo structures. And I'm going to call it as K15 Now, there are various formats available when you want to save structure. As you can see here, the most commonly used format is .mol. It's a MDL mol file. And also another format which is readable by many of the molecular docking softwares or the other softwares is the SDF, .sdf, which is MDL SDF file. These are two very commonly used formats. But there are several other formats like proprietary format from CameDraw, which is .cdx. So when you save structures from CameDraw, generally they get saved in the CDX format. But many of the open source drug discovery tools, they do not read these CDX formats. So it is always essential that you save those structures in correct format. The most commonly format, as I said, are MDL mol file and MDL SD file, SDF file. So I'm going to go ahead and save this structure as the MDL mol file. So I have saved it. I will go to that folder now. It's on the desktop. Demo structures, you will see that there is a mol file available. Now, if I want to open that structure again, and now what I will do is I will go to let me delete these structures. And if I want to open it, I will just go to the file section, open, go to the demo structures. You will see here the structure is there. It also shows you preview. You click open and you have the structure available. So this way you can save the structures and you can use those with any other softwares. So I hope you have got at least slight idea about how you can use these softwares to draw complex structures. You can obviously use some of the templates which are available on the left hand side. You can use these different elements to introduce different heteroatoms in the molecule. You can also use the very simple tool which is convert name to structure to generate drug molecules, which will save a lot of time. Obviously, there are a lot of arrows available. Let's say if I want to show some reaction, I can use those arrows. But I think it makes no sense showing all these tools right now because what we need to understand right now with this demonstration, how to draw structures and how to save those files and how you can interconvert those in another format. So I think with this, I will stop my demonstration on the K-Maxon for now. If anyone has any doubt, you are free to ask me. Is there anyone who has any doubt or question regarding the K-Maxon? There are so many things to explore actually, which I have not shown all the things. Like I told you that if you draw a structure in very haphazard manner, like for example, let me do it just on purpose. Now you see that this structure which I have drawn, it's not really drawn properly. The bond angles, bond lengths, they are not good. So there is a one tool which I told you, which is structure cleanup. So what you have to do is you have to select such structure which you want to clean up, go to the structure, and there is an option here which is clean in 2D or clean in 3D. Clean in 2D is fine because we are drawing 2D structures. I click the clean in 2D. You can see that the structure which I had drawn was really very badly drawn structure, but now you can see that the structure which is drawn, it is very much representable. Now you can copy these structures and paste in any of the programs. So let me show you. I'm going to open a Word document. And I'm going to copy one of the structure. Let me copy actually Gefitin structure, Control C, and simply use the Control C, Control V command I'm sorry, in K-Maxon it is a bit tricky. So I will have to select the structure, go to the edit menu, and then you have to go to the copy. And then you can copy it as a CameDraw file or the Marvin doc tool if you want to copy just the hello hello okay मी चेक करतो कधी गेला आत्ताच गेला का I'm sorry, I think there is some network issue. I got message from Dr. Ashish. Let me try switching my network. Just give me a moment.
Yes, Neha, do you have any doubt? Neha, you have raised your hand. Want to ask anything? You can unmute yourself and you can ask your question. Okay, so for molecular docking, we need structures in different format, like mol2 format, and sometimes we need to convert the structures in PDB format. Okay, so Dr. Kapil is here, so he will explain you how to convert those structure. So over to you. You're mute. Yeah.
I'm sorry, everyone. There was some issue again. So let me share my screen back. So what I was talking about was how you can use these structures and copy those in different formats. So let's say I want to copy it as a MRV format. I can just click on it, go to the file, but when you put that, it will be added in some code format. So it is better if I select this structure and copy it as, I think if I copy it as CDX, it should be able to. Or if I copy it as an image format, the problem copying it as an image format is that the structure becomes uneditable. So obviously you can copy it as an image if you don't want to edit these structures further, but it is advisable you copy it as it is so that the structures can be edited later on. So this is how you can use the KIM Axon software to draw structures, to clean those structures, to convert the name to structure, or to calculate different properties. Now, as Dr. Ashish said, now I'm going to show you how you can convert one format to another. Obviously, KMaxon allows you to convert structures into other formats, but those formats are not all formats are useful when you want to use, for example, Autodoc, which Dr. Ashish is going to use in the next lecture, next demonstration tomorrow. So let me show you which the software which you can use for converting the structures. One of such software is OpenBabel. So you can just search OpenBabel on internet and you can download this software. It is again free for use. So when you go to this software, let me open it. So you can see that you have two windows, one on the left hand side, one on the right hand side, which is the left hand side one is the input window. On the right hand side, you have the output format. So here you have to select what is the format of your input molecule. You can see that so many formats are available. So there are n number of formats. You can convert any format into other format when you using the OpenBabel. So now I had drawn the structure of gefitinib in using the mole format. So I'm going to select MDL mole format. I'm going to select my structure, which was drawn just now. So let me go to the desktop, demo structure. So here is my gefitinib molecule. So it shows you your molecule in the mole format, but obviously it shows all the coordinates in the text file. Now I want to convert it into the PDBQT format. Why I want to convert into PDBQT? This is the format which is read by Autodoc. Autodoc uses the format which is PDBQT. So you can see again, if I want to convert this structure, there are so many formats are available. I can convert it to the ChemDraw format to the mole, SDF. So you can see so many formats are available here, which can be easily used. So now I want to convert it into PDBQT format because that is the one which is used by the Autodoc, which Dr. Ashish is going to use in the next session. So here I'm going to select the output file. And let me give the name again, gefitinib name. Now, if you have multiple files, you can also do that. For now, I'm showing you the single file. You have to deselect output below, because if you click the output below, only the textual matter will come down here, and then you will have to manually save it. So instead, just unclick that. And then what you have to do is you simply have to click on the convert button, which is present here. There are several parameters, obviously, which you can be used depending on the what type of format you are converting into what type of format. But for now, when I'm converting the mole file into the PDBQT format of the Autodoc, I'm not going to select any of the options which are additional options which are given here, but they are useful when you are manipulating many files and you want to make any changes when you are converting. So you just click on the convert button here, and you can see that it gives you the message also that one molecule is already converted. So this tool is very nice, OpenBabel, when you want to convert in any format to any format of the structure. So sometimes you get stuck that the structure which you have drawn with the help of Kmaxon or ChemDraw, it is not readable by the other softwares. So you can easily convert those structures using OpenBabel. Now let me go back to the folder where I have saved it. So I will go to the demo structure. Now you will see that there is a PDB-QT file. Now, obviously, this PDB-QT file is not readable by any softwares except it is generally readable by the obviously Autodoc, and there are some other tools which can read such PDB-QT output files also for the structure analysis, which I'm not going into detail because Dr. Ashish is obviously going to show you all those things related to the Autodoc in the next session. So this is how you convert one structure into another. I think Dr. Ashish will use this software also to convert the structures and to use those further for Autodoc Vina. So let me go back now to my presentation. The next part which I'm going to talk about is Data Warrior. So if there are no questions regarding the Kmaxon demonstration, I'm going to show you the Data Warrior and how you can use this tool for optimizing the lead molecules, how you can optimize molecules when you are in the drug discovery program. So before we go to that, let me just discuss about what is Data Warrior and what are its application and some general information. So it is an open source program for data visualization, analysis, and it has some chemical intelligence as well. So it was designed to help drug discovery community to visualize and analyze different relationship between chemical structures and biological data. It was developed by Actelion Pharmaceuticals, and released in 2014. Now the current version is 5.2.1. There has been a lot of development. I'm using this tool since 2000, I think 17, if I'm not wrong. So almost 3 years I'm using this software for analyzing and visualizing the chemical structure as well as the biological data. One advantage of this tool, again, its license is free. You have this tool available for Linux, the Mac, as well as for PC. So everyone can use this software, and you can get it from this openmolecules.org website if anyone wants to try this software. What are the different features? It has interactive data visualization and analysis. It has built-in chemical intelligence. You can do a lot of calculations. You can do real-time data filtering. So I'm going to use all these things when I'm going to show the demonstration. I'm going to show you the visualization. I'm going to show the calculation of different properties. I'm going to show you the data filtration. It also has dedicated chemoinformatic module to support drug discovery. Apart from that, it runs on, as I told you already, it runs on Linux, Macintosh, as well as on Windows. And so if you have the computational demanding algorithm, it is also able to use all the cores of the processor. So the calculations are faster. What you can do with this tool, you can do the data analysis, chemoinformatics, you can calculate different descriptor, you can use different databases. Now this is amazing tool which is available in Data Warrior. I'm going to show you in the demonstration where you can retrieve structures from different publications which are listed on PubChem. So you don't have to draw the structure. Let's say I am working on certain target. I'm going to use one target for the demonstration purpose. But let's say if you're working on certain target and you're doing drug discovery and you want to retrieve all the structures which are available in a publication, different publication, you just have to give the PubChem IDs and you can not PubChem, sorry, PubMed IDs, and you can retrieve all those structures with a single click. Very nice tool. It has other databases like Wikipedia structures. Like we are not going to use all the information from Wikipedia, but at least the structures which are available on Wikipedia, there are around 18,000 structures available. You can download those easily and you can import those without drawing any structure. So I'm going to show you that also. You can do a lot of automation with the help of macros, which you can figure out. Uh, there are so many things you can do with this software. So obviously, because of the time limitation, I'm going to show you the demonstration to how you can use it to optimize your molecules. And it can read data and structures from spreadsheet files if you have, which has a smile strings. As we have seen, the smiles can be converted to structure. With this software, you can have a spreadsheet with a lot of smiles present, and you can just import it. It will convert it to structures directly. You can also use standard SDF files. And as I said, it has a database, so you can directly import structures from Campbell, which is a huge database of structures which we are going to use today. And you can also import structures from Wikipedia molecules. So for today's demonstration, I just want to give you some background. Now this demonstration, more information if you want to have, it's available on the web page, which is given down here. You just have to go to the mmv.org, which is an open source research development for the malaria. So I came to know about it because I'm working with the Drugs for Neglected Disease Initiative, which is an open source drug discovery platform, which works for the neglected diseases. So they are also working with these softwares to develop tool for the drug discovery, which are free for everyone. So I'm using the demonstration which was actually we had used to understand how to use this software, but uh, hopefully you will understand how you can use this data warrior for optimization of molecules. So what we are going to do is we are going to use ligand efficiency matrices. So there are 3 different ligand efficiency matrices. I will talk about it later on to optimize the CCR5 antagonist. What is CCR5? Is nothing but CC chemokine receptor type 5, which is used as a target in the anti-HIV drugs. This target is actually important for the retrovirus to actually get inside the cell. And this target is if you block it, if you use antagonist, the viruses do not get access to the cells. So what we are going to do is this demonstration will show you the real medicine chemistry analysis of the properties of CCR5 antagonist. And we are going to use one drug, anti-HIV drug, which is available in market, which is Maraviroc. And we are going to compare different antagonists which were developed by different companies. And we will see how the drug discovery projects of different companies, where they stand as compared to the drug which is already available, and how we can use it to optimize your the molecules and further do use it in the drug discovery. So what we are going to use different free tools. It includes the PubMed and Campbell to assemble the data set. We are going to import the structures which are published by different pharma companies in publications. Then we are going to use Data Warrior to visualize the relationship between the ligand efficiency matrices. I will talk about it, and the molecular weight for each series. And we will highlight choices made as molecules were optimized towards drug. Okay, so the workflow for our demonstration would be, I'm going to use PubMed to find out the literature which contains these ligands. We are going to use 4 publications from 4 different pharma industries which we have worked on this anti-HIV target. Then we are going to use Campbell with the help of these PubMed IDs to extract the structure as well as the biological data. So we will get the structure, we will get the IC50 values. So you don't have to do any typing, you don't have to do structure drawing. We are going to import hundreds of structures. We are going to filter those structures with this software very easily. And with the Data Warrior, we are also going to analyze different properties of this published compound and calculate new properties and visualize the data, which can help us to optimize these molecules further, which would be useful to identify new drug molecules. So what we are going to exactly achieve in this optimization is that you must be knowing that the physicochemical properties of molecules are very important for the quality of drug candidates, and analyzing discovery compounds that have made to the clinical use, or which have failed those clinical trials, or they were not successful, can be used to understand what makes a good drug. So there are two major challenges: identifying a good chemical starting point, which we generally call as lead molecule, and optimizing that molecule while having the balance between the in vitro potency as well as the physicochemical properties, which inhibits the bioavailability like ADMET, like absorption, distribution, metabolism, excretion, and the toxicity of those molecules. So we need to have the balance between those. What happens is, in many of the drug optimization programs, one looks at the in vitro activity, one tries to improve the activity, which may lead to molecular obesity. Now, in many cases, how one increases the in vitro activity is by increasing the lipophilic substituent, lipophilic components in the molecule. So increasing lipophilicity and molecular weight, in many cases it does increase the in vitro activity, but the problem is that it decreases the drug like properties. So we have to find out a balance between the increasing the in vitro activity while maintaining the drug like properties. So we are going to do that with the help of the data warrior software. Now, as I said, there are three parameters which actually uh, give us the ligand efficiency matrices, and these parameters are designed to guide medicinal chemists towards the most drug-like compounds. And these are, one is the LE, which is ligand efficiency, which is calculated from the IC50 values, and so you have a formula which is IC50 binding energy, and it uses the log IC50 values to calculate this ligand efficiency. And we generally should aim for ligand efficiency which is more than 0.35. That means higher the ligand efficiency, better would be the ligand. Then you have the lipophilic ligand efficiency, which takes care of the lipophilicity, because there would be generally loss of bioavailability if there is an increase in the lipophilicity, if there is a too much increase in the lipophilicity. So the LLE generally should be around more than 3 for the lead compound. It should be more than 5 if you have a clinical candidate. So this can be also calculated using the data warrior software. And lastly, we have the ligand efficiency lipophilic price. That means how much is the ligand efficient and how much is the price we are paying for the increase in the lipophilicity. And it should be around 0 to improve the potency. So we are going to use these three parameters to understand or to optimize the molecule, to visualize the known molecules so that we can optimize this further. If you want to know further about ligand efficiency, I would suggest you to go through this paper in the Drug Discovery Today. It's from the Sabine Schultes, and it talks about the ligand efficiency as a guide in fragment heat selection and optimization. Very nice paper. So please go through this so that you can understand all these three parameters which I've talked about and which we are going to use to optimize molecules. So what is the link between the lipophilicity and polypharmacology? That means there is one more problem. When you increase the lipophilicity and when you have smaller molecules, the chances that it hits more than one target increases. Now, we generally do not want our drug molecules to hit more than one target, which may lead to toxicity. So many drugs, if you see the left-hand side image, they have more than one target. So there are only 15% of drugs which have only one target. There are more than around 50%, 35% of drugs which have more around 2 to 5 targets, and rest 50% of the drugs are the one which have more than 6 protein targets, which are generally defined by Ki, Kd, IC50, VC50, which is less than 10 millimolar. So you can say that many drug molecules, they have more than one target, which can be a problem. So when you're doing the designing of the molecule, you have to take care of the lipophilicity as well as the size or the molecular weight. The right hand side image gives you the idea about it's a comparison between C log P, molecular weight, and the mean promiscuity, which is nothing but the possibility of that molecule to hit more than one target. So as you can see, if the log P is too high and if the molecular weight of the molecule is small, the chances that it hit more than one target increases drastically. So small lipophilic compounds are likely to hit large number of biological targets. So with the help of this tool, the data warrior, we are also going to understand the relationship between the C log P and the molecular weight and how we can use that to optimize the molecules further. So we are going to use 4 data set, as I told you. One data set is from the Pfizer, which actually identified the drug, the Maraviroc. Then we are going to use data set from the Merck. The third one would be AstraZeneca, and the next one would be GlaxoSmithKline. Now they have published their work in different papers. Pfizer has published in Bioorganic and Medicinal Chemistry Letters. Merck is also, I think all of them are in the Bioorganic and Medicinal Chemistry Letter, the 4 companies, but they have worked on very similar molecules. And out of those, the Maraviroc, which is given on the right-hand side, is in the clinical use for the HIV. Now, what we want is that we are going to need the PubMed ID. So the first step we will have to do is that go to the PubMed, search for this reference, which is given here, and copy the PubMed ID, which I have done already for you. So AstraZeneca has this the paper has this PubMed ID, the Merck has this PubMed ID, Pfizer and GlaxoSmithKline. So these 4 PubMed ID we are going to use to import the structures from all these 4 papers. So there are around 300 structures altogether from these 4 papers, which we are going to use. So to retrieve these structures, as I told you, we are going to use a database which is Campbell. This Campbell is an open source database. It is a manually curated chemical database of bioactive molecules with drug-like properties. So you don't have to go anywhere searching the databases of the structures. You can import not only structures, but you can import the chemical properties as well as the biological activity of those molecules. This Campbell database is maintained by European Bioinformatics Institute of the European Molecular Biology Laboratory. It has around 2.1 million compounds. It has a data of 1.4 million assays. You have 13,000 drug molecules, 80,000 documents. You have the data of 2,000 cells, different cell line studies. You can see that you have 14,000 targets. So you can select targets also from among those to import those structures. If you know the target upon which you are working, you can do that as well. So now this is just the introduction of what we are going to achieve in today's demonstration. So I'm going to now open the Data Warrior, and if you have any doubt, please let me know. All right, so let me give just brief information on Data Warrior. This is a simple window. There is nothing actually. If you want to create a new file, you can go to the file, new, and then you have to add columns. Like I want a text column. Let me call it as the name of the compound. I will add this. Let's say I want second column, which is the structure of the compound. I click OK. So it adds these 2 columns. And if you wish to add any new rows, you can do that. So I'm going to close this. So you can see that now you have the name and structure. And if you want to add any compound, you can do that. For example, I want to add gefitinib. I can type there gefitinib. Okay, so it adds the name. And if I want to add structure, I can double click there. So you have to double click to edit any of this field. When I double click, I get the editor. Either you use this editor or you can use the structure which you have drawn earlier. For example, I had drawn the structure using my Marvin sketch. So I'm going to open that structure. I'm sorry. Let me open it with Marvin sketch. So I have this structure drawn already. I have just drag and drop. I'm going to copy it, Control C, and I go here and I paste it. So you don't even have to, if you have used the Chem sketch, you have the structure drawn, you can simply copy paste it here, click OK. Now you can add the structure. Now, obviously you can add more columns, like if you want to have IC50 value or any other parameters, you can do that. You can do a lot of modifications, or you can do a lot of calculations. You just have to go to the chemistry here. And from the chemical structure, you can calculate different descriptors. You can calculate different properties. You can calculate smile code. Let's say I want to, I have a database of 100 compounds. I want to create smiles. I just click add smiles and you create a new column. You have to select what is the structure column. I have given the name structure. So the new column will add it with the smile. So you can see that there is a new column added with the smiles. Very easy. Let's say if I want to calculate something else, I want to calculate different properties. You can see you have different properties for the drug likeness. So this is very much important for the drug discovery projects. You can calculate the average molecular weight, you can calculate the log p values, you can calculate how many hydrogen bond acceptors are there, hydrogen bond donors are there, you can calculate the total surface area if you need a total polar surface area, which is very commonly used in the QSAR studies. You can also calculate I will go to the Ailey Tox shape later on, but you can also calculate other things like functional groups, ionization. For now, it is not needed. So you can, if I click OK here, you can see that there are a lot of columns which are added. Like you have the, just a moment, this part, let me move it down. Okay. So you have like total molecular weight, automatically calculated, C log P, hydrogen, how many hydrogen bonds acceptors are there, how many hydrogen bond donors are there, what is the total surface area or total relative polar surface area. You can calculate that everything. Amazing software, because you can you can import structures and you can calculate these properties very easily. Now let me go to the importing part. So you have here database tab, you go to the database, you can see here retrieve Wikipedia molecule, search Campbell database. We are going to use this today to understand the CCR5 antagonist and to optimize how we can use this tool to optimize the molecules. Before that, let me show you the retrieving Wikipedia molecule. If I go there and if I just click there, it is processing it. Depending on the net speed, it will take some time. Now you can see that it has imported how many structures. There are 18,263 drug molecules, or the different molecules which are in the Wikipedia database. Now let's say if I want to draw any structure, I don't really have to draw it. I can just either look into the list or I can search it very easily. You can see on the right hand side there is a field to filter the compounds. Let's say if I want to use gefitinib, I will just type gefitinib. Oops, spelling was wrong. Sorry, it's gefitinib. You can see that it directly filters the structure. I can simply copy it, the structure, and copy as a structure, 2D structure, and paste it anywhere I want. So like I have copied it, I paste it here. I will have gefitinib structure directly imported from the Wikipedia. So this is very nice tool because you have a databases of lot of structures from the Wikipedia directly available, and you can filter those very nicely. I'm going to use this later on to import one drug molecule. So I'm going to keep it open. I'm just minimizing it. Now I will close this file and I will go to the demonstration of optimization. And I think I just closed everything. All right. So what now we are going to do is we need to have the database, as I showed you in the my slides. We need to import structure from these 4 publications, one from Pfizer, one from Merck, one from Glaxo, and one from AstraZeneca. So I'm going to copy these PubMed IDs, which I have copied already because I don't want to type those. You can type those. And you have to put those with one space in between those numbers. These are just PubMed IDs. I have copied those. Now I'm going to go to the database again, and I'm going to click on the search Campbell database. When you do that, it opens a window. Either you can select targets. There are so many targets available. You can filter those targets. But now my plan is to import the structure from those 4 publications. So here you have downfilled PubMed IDs. So I'm going to put those PubMed IDs here with one space. I'm just going to click on the OK. Now you will see that it imports around 321 structures. Sorry, 320. Yeah, I was close. So you can see here there are 320 structure. Let me make it bigger and also zoom this window. You can see that it has imported structure from 4 publication, and you will see that it has a lot of information. It has the IC50 or IC90 values. So you have the value, the unit is nanomolar, and type. You can see for some of them it has T-half, some of them IC50, some of them it is IC90, area under curve. So you have a lot of biological data here. It also gives you what was the target if it is was ADMET target, admit target, or it was the target which we are looking for, human immunodeficiency virus, or the target which actually we are looking for is a CCR, which is a CC chemokine receptor type target. So we are going to use this target on only the data for that target, and you have the references as well as the PubMed ID. All right. So what I'm going to do now is I'm going to filter these molecules because I don't want all data. What I'm interested is in the IC50 values against CCR5, because we are looking at the CCR5 antagonist, and we are going to optimize these molecules based on their IC50 values which are reported. So I'm going to filter that. So to filter any data, as I told you that the data warrior can be used for filtering a lot of data, I go to edit, I create a filter, but before that I want to remove all those filters which are present earlier. I click on remove all filters, and then I go to edit. Here I go to the new filter. Now I want to filter it based on the IC50 values. I just want the structures or the data where the IC50 value is given. So I will go to the type. You can see the column is type, which has T half, C9, IC90, IC50. I will go to the type and I will select the text one filter. So now we have here on the right hand side that filter. I'm going to type there IC50. Now you can see that it has filtered the molecules, only those which has IC50 value. I hope it is clear to all of you, right? Now, the next thing I want to filter is the based on the receptor or the target. You can see that it is not only one target. You have the voltage gated calcium, HERG target. So different targets are there. I'm not interested in all the targets. I'm just interested in the CC chemokine receptor target. So to filter that, I'm going to copy first the target ID. Now the target ID is given somewhere here. Let me just check. Sorry, here. You can see there is a target ID given. I'm just going to copy that target ID because I want to filter it with that. So again, I go to edit, create a new filter. I go to the target ID and text. So I'm going to filter it with the target ID number, and I'm going to put that number which I just copied. So what happens is it removes all other targets and it keeps only the target which I'm interested in. And you can see that how many number of molecules are there, 91. So we had 320 molecules. Out of those, we have filtered the data which is we require of 91 molecules. Now I'm going to save this as a separate file because I don't want the rest of the data. I just want this data for my analysis. I'm going to go to the edit. I'm going to go to the data. Wait a second. Go to the edit. I'm going to select all molecules. So click on the select all and go to the file, and then you have to save it separately as a new form. So I'm going to click on new form, and I'm going to click on the selected rows. So I have selected the rows which I have filtered. I'm going to click it. It will create a new file only with 91 molecules. You can see that there are 91 molecules only which we needed. So I'm going to close the earlier file which we don't need anymore. Oh, by mistake, I closed the file which we needed. So I will again generate it. I'm going to close the earlier file. All right. So now I have the file with filtered data. I have 91 molecules with their IC50 values, everything, and you can just have a look at it. Now, one thing which is missing in this data is the drug. The drug. What was the name? It was let me check. It was Maraviroc. So we need to import this drug because this drug is not present, and this drug has a IC50 value of 77.2 nanomolar. So what I'm going to do is I'm going to create a new row because I want to add that drug molecule which is not present in the publications. So I'm going to go to the data. I'm going to create a new row. So I'm going to click on add empty row here. So I would just want one row for addition of that drug. Now, if you just look at there, so you can see that row here. Now I want to import that structure. How to do that? Now I'm going to use my Wikipedia database. I will retrieve that Wikipedia database from database and then click on the retrieve Wikipedia molecules. I'm going to write here Maraviroc. So even I write partially, you can see I have got the structure of Maraviroc. It's very complex. I don't want to draw it. I can simply copy it from the Wikipedia source. I right click on it and click on the copy structure as 2D structure. So I'm going to copy that structure, go back to my file, I double click here, it will open the edit structure menu, I paste it there, click OK. Now we have the structure available. Now what is missing here is we don't have the IC50 values. We know that it is 7.2 nanomolar. So I'm going to add it manually here. So value, double click there. I'm going to put 7.2. Then it is nanomolar. So I have to give the unit. And then it's IC50 value. So I'm going to write here IC50. So now we have the database ready with their IC50 value. What we need to calculate now is the molecular weight of this compound. As I said, you can calculate different properties for all the molecules. So now I'm going to calculate the molecular weight of this compound. So for that, you have to just go to the chemistry, then select the from chemical structure, calculate properties. So I'm not going to calculate all these properties. We don't need these properties. So what I'm going to calculate first is only the molecular weight, and then those three parameters which we are going to use. I told you we are going to use those three parameters for the ligand efficiency, LE, LLE, and LELP. So I will go to the LE tox shape. You can see here we have the ligand efficiency, we have the lipophilic ligand efficiency, and we have the ligand efficiency lipophilic price. Now these would be calculated based on the inhibition data. Either you need to have the Ki values or IC50. We have the IC50 values. So I'm going to select value. Value is nothing but the column which has those IC50 values. You can see here it's a value which has those IC50 values. So I'm going to select value everywhere. Click OK. It will generate those properties for all the compounds. So I'm just going to go right hand side. And now you will see that it has added 4 new columns. Total molecular weight. You have the LE from value, you have the LLE from the value, and you have the LELP from the value. All right. Now let's start with the analyzing these values, how you can analyze these values or visualize these values, which would be useful for optimization. So to do the visualization, which can help us in the optimization of molecules, I'm going to right click on the table bar, and you will have the new 2D view. You can visualize it. So I'm going to click on the 2D view. Obviously the axes are not correct. So I'm going to change the axis. I'm going to keep the molecular weight on one of the columns. So it's a total molecular weight. And on the second one, I'm going to keep PubMed ID. I'm sorry, not PubMed ID. It was right, LLE. I'm sorry. We have to have, yeah, LLE on the second one. You can see that you have all the molecules which are scattered based on their total molecular weight. So you can see all the molecules are in the range of around 350 to 650 Dalton, and you have the different LLE values. Now, if you look at this, if I go back here, you want to have the LLE values more than 3 or more than 5. So by visualizing this, you can filter out molecules which have the LLE values lower than 3. So you can see any molecules which is down below that 3, those are actually not good values. So if you click on that structure on the right hand side, actually you get that structure in detail. And you can see that what are the properties of that structure, and you don't want such structural features in your molecule because it has lower LLE values. So this way you can visualize and understand which type of molecules you want and which you don't want. Now, to make it easier, I'm going to divide this diagram into publication, or the AstraZeneca one separately and the Merck one separately. So for that, I'm going to right click on any of the circle. You go to the set marker color. So there is something called set marker color. I'm going to click on that, and I'm going to click assign the marker color based on the PubMed ID, because that's how it differentiate which one is the AstraZeneca, which one is Merck, or which one is Pfizer, or the GlaxoSmithKline. So I'm going to click on this. Now you can clearly see that each of this company has worked on very different kind of data set, different kind of properties. You can see on the left hand side, this greenish yellow colored, these are actually from the PubMed ID 1678236, which belongs to, let me just check, Pfizer. So Pfizer has worked on the molecules which have the molecular weight less than 500. You can clearly see that their molecules were basically having the molecular weight less than 500. If I check the red one belongs to the Merck. Now, if you see the Merck one, they have the molecular weight in between 480 to around 550, and all of them generally have the LLE value higher than 4. You can see that they have LLE values higher than 4, so you can select those. You can understand how the Merck has worked. The blue one is from, let me just check, it's from the GlaxoSmithKline from the PubMed ID. So the GlaxoSmithKline has really worked on the wide range of molecular weight, but many of their compounds have lower LLE value. That means those compounds were not really good. And the right hand side, you may have the yellow color structure, cluster, which belongs to the last one, AstraZeneca. And their structure, you can see that they have very high molecular weight, and many of those have lower, some of those have lower LLE values, but many of them have higher LLE values as well. So now, why this tool is useful for optimization of molecule? Now you can understand that what are the structure which have the undesirable properties, what are the structure which have the desirable properties, and you can then do the optimization of those molecules further by studying those structures and what are the properties which you don't want to introduce. And here you can see a gray colored dot, that is actually the Maraviroc, which is the drug which is available for, and which is anti-HIV drug available. Now, if you want to split these structures, I don't want it same diagram, I can also do that. You just have to right click there, and then you can split the view by the categories. Now, what are the categories? One is from the Merck, one is from the Pfizer's, GlaxoSmithKline. So I can click there, and then I want the first column to be PubMed ID. So you can see that it simply divides into 4 different diagrams. Now it is very easier to understand and visualize the data, where the data from the red one, as I said, the Merck is completely different data. You have the second one, the yellow color, which was from the, let me check, AstraZeneca. So you can very easily understand and visualize the data, which is useful for the optimization of the molecules. So this is one way you can do that. There is another way by which you can optimize these molecules further, is by using 3D diagram. Now, I hope you have understood using this diagram, using these charts or the graphs, we can easily visualize and understand where those molecules are lying, or if they have the optimum properties or not. And you can select the molecules which have the optimum properties, and you can further optimize those by introducing the desired characteristics. So this is very useful for the drug optimization when you are in the drug discovery program. Let me try to show you another way of showing this data. I'm going to now create a 3D view. So I'm going to create a 3D view. I need to change my axis. So I'm going to click on the XYZ. The first one I'm going to use is LE values from the which we have calculated. The second one I'm going to take is LLE value, and the last one I'm going to take is the molecular weight, total molecular weight. So you can see that you have the 3D diagram. Here also I can divide those molecules now by using a marker based on the PubMed ID. So now you can clearly see that where the clusters, there are 4 clusters for the molecules from the AstraZeneca or the GlaxoSmithKline or the Merck, and you have the gray colored dot, which is actually for the Maraviroc, which is actually sits in between. So you can, it's not necessary it has the most optimum properties, you can further optimize those. So what we wanted was LE value should have been more than 0.35. So you can select the molecules which are on the right hand side here. You also want the the LLE value should have been the more than 0.35. So you want it somewhere here. And the molecular weight, it should not be too low, because we have seen that if you have the molecular weight too low, high lipophilicity, there are chances that such molecule will hit more than one target. So you can select the molecules which are of slightly higher molecular weight. So again, using this 3D diagram, you can visualize the different properties or the different parameters which we have calculated. So So this is one more way you can generate a figure. Now let me try to show you another way, which is using bar diagram. There are some papers publication available which you can go through, which uses the bar diagram to again compare these data sets. So I'm going to go to the 2D view, and I'm just going to change the type of diagram which is shown here. So I'm going to first make it just one diagram. So we had split it into 4. So I'm going to change the parameter to unassigned. Now we have the single image. Now I'm going to convert this into the bar diagram. To do that, I'm going to right click here, and you have to select set preferred chart type. And when you click there, you get different types of charts. I'm going to select bar chart, and the bar size or pie size I'm going to select based on the molecular weight, the mean value of the molecule. So here you have to select the mean value, and here you have to select the total molecular weight. I'm going to click on OK, but one more thing we have to change is the XYZ. So here we are going to select PubMed ID, and in the down one we are going to select unassigned. Now you have a very nice diagram which shows you in each publication how the molecular weights are differing. You can see that the greenish yellow color you want to say that one belongs to the Pfizer. So Pfizer has the lowest molecular weight, mean molecular weight of compound, while the yellow one, which belongs to AstraZeneca, has the highest molecular weight. So you can actually compare this molecular weight with the biological activity, and you can select the optimum, the best molecule for the further optimization. So this diagram is also very useful to understand how the different data sets are lying based on their molecular weight. Now what we can do is that I can change the parameter to log P also. Let me check if I have calculated log P or not. I think I have not calculated log P. So what I will do next is I'm going to calculate the log P value of this compound. So simply I will go again to the chemistry. From chemical structure, I'm going to calculate properties. And here I'm going to calculate C log P, which is octanol water lipophilicity of the molecule. So I click OK, it calculates, it creates a new column. Here you can see that there is a new column, C log P. Now I will go to the 2D view again, and now just I'm going to change the parameter. So I will go again to the set preferred chart type, and instead of total molecular weight, I'm going to select C log P. You can see that now from these papers, I can also compare the log P values. You can clearly see that the red one, which belongs to the Merck, has the lowest log P values, while the one which is blue from GlaxoSmithKline has the highest log P value. So when you want to have the optimum log P value, when you don't want to have a very high log P value, you can select the correct database. Now, when you're analyzing all these data sets, now obviously when I'm visualizing these 2D structures, let me create one more graph. So we had seen that I had created this PubMed ID. Created this graph for LLE. You can create also for the LE. You can also create the similar graph for LELP. So you can, depending on those parameters, you can study all those three. I'm sorry, depending on the graphs, you can study all those three parameters, which are essential for the very important for the optimization of the molecules. And when you visualize this using this software, you get more insight into the molecules which are already reported, so that it can help you for the further optimization. Now, in general, when you are doing this optimization, you do not want to select only one criteria. What will happen is if you select just LE, for example, now let me change it to LE. If I just focus optimization based on LE, what will happen is that there will be a matrix bias, and it will lead to generally smaller molecules. Let me check here. So we have the, okay, let me change the total molecular weight, and I want LE here. So if I go for the LE now here, that higher LE is fine. It's good for my molecules, and I'm not looking at other two parameters. I'm just focusing on one parameter. But the problem here is the the compounds having higher Ale, they are not necessarily having higher molecular weight. They are of the lower molecular weight. We have already seen that the molecules which have lower molecular weight, and if they have the higher log p value, they have a chances hitting more than one molecule. So if I just use Ale, that would be a quite biased selection. I should always visualize and analyze all three parameters, Ale followed by LLE. I should find out which are the optimum one for optimum properties of those compounds, which can be useful for optimization in the drug discovery. So I hope you have understood that how I can use this software, with amazing software, to not only create database, to import structures from different publications which are listed on PubChem, and generally the PubMed publication. These structures are generally available in the Campbell database. So what we have used today, we have used the data warrior software. First, we imported structures using the database, using the Campbell database. Then we added the missing structure using the Wikipedia molecule, which was the drug molecule. Then we calculated different properties from the chemistry. And then using those properties, we generated the graph. And obviously before doing that, we also did some filtration. So by going into the edit, we created new filters, and that way you filter the data so that you have the only desired data which can be used for the visualization of different properties, which can help us further in the optimization. So with this, I think I will stop with my demonstration. So before I stop, I just want to thank my funding agents, especially DNDI, because of the DNDI, I came to know about the Data Warrior, which is we are working on a project of Lead Optimization Consortium of India, along with IIT Gandhinagar. So we have funding from, so I had received funding from SERB as well as DNDI and from NMIMS, and we have a collaboration with IIT Gandhinagar and DNDI. I'm really thankful to the NMIMS for giving opportunity, and this is my small group. Out of these, the Utkarsh and Suchita were working on the DNDI project. They have now left, and there are two new students who are working on this project. With this, I would like to stop, and I would be happy to take any questions from the audience.
Thank you very much, sir, for such a wonderful presentation. Now the session is open for question and discussion. So if you have any question, anyone, please ask. You can drop question in the chat box as well.
I see one question.
But that is partial. I think it is not complete. If we have designed molecules and don't.
I think question is incomplete. I don't understand.
Geeta, can you please type this question again, or if you can unmute and ask it directly. So I have one question. Can we export this data in different formats?
I think you can. So you have to go to the, so once you have generated this database, you have to go to the file, and then you have the export option, I believe. Generally, we use the format of the Data Warrior only, but I think you can save it as SD files. Yes.
SD file, yeah.
You can save it as SD files, and which you can then import in other softwares.
That's true. There is one question. Can we go with our design molecules? Can we analyze with Data Warrior?
Yes, you can. So if you have the database created of those structures, or even if it is not ready, you can draw those molecules with the help of either the tool which is already present here to draw the structures, or you can make those structures using Chemaxon, Marvin Sketch, or any other tool which you prefer. Create the database, and you can analyze those molecules. You can calculate those properties. If you have those IC50 values, you can create. One easier thing would be you create to make an Excel sheet with the headers, whatever you want, and then import it, because it becomes difficult to type in here. You have to always double click here. So it would be easier. You create an Excel sheet and then import that file here, which can be done quite easily in this software. So yes, you can go with the design of your own molecules. You can analyze your own molecules in the Data Warrior.
there is similar type of question from Gita. If we have designed molecules and don't have any database, then how can I use this software?
I think it's a similar question. So you will have to create your, if there is no database available for certain target, you can create a database in Excel, for example, with the structures and the properties. If you have the IC50 values or any biological data, you can create and import it, or you can create the table itself here. So that is possible. But I would suggest you just go through the database of the Campbell, and there are so many targets available. It may happen that your target is listed, it's one of the target, and you can just simply import those structures. There are so many structures available to actually do the chemoinformatics, and this tool actually helps you to import all those structures so easily.
Yeah. I would like to add one more thing in this to answer this question. If you have your own designed molecules and if you don't have any comparative database with you, so you can go for Tanimoto similarity study. So you can select the marketed drug molecules. You can identify the properties and you can compare the property-based similarity. That is Tanimoto coefficient, and from that you can easily compare your molecules with that. So any other question from any other participant? Okay. So thank you everyone for joining today's session. We'll post feedback link to you in chat box. I guess yes.
I'm yeah.
Yeah. We'll
I'm going to
share.
post the feedback link. It is requested that everyone please uh, reply to the feedback. Just give me a moment. I'm doing that.
And somebody asked in the morning session that will get certificate. Yes, you all will get certificate. Those who have participated, and from the feedback, we'll collect the data, and within 1 week, we'll share the certificates with you all.
There is one question: is there any manual to use this data warrior? There is a manual available on the data warrior website. So just go to the openmolecules.org. You will find a manual available there, and there are a few videos also available if you wish to. Uh, uh, the video recording is available for today's session. Just give me a moment. I will give you the link to all of you. It would be available on the YouTube. So it has been already streamed on the YouTube. So please follow the link which I am pasting in the chat box. You can use this link to watch today's complete session. I may remove the gaps which were there in between, which will take 1 or 2 days because there were time slots. Okay, I have to open the Google form. Just give me a moment. Just give me a moment. I'm sorry. I have not opened the Google form. Just give me a moment.
Okay, so while Dr. Kapil is working on the Google form, tomorrow we'll start at 11 a.m. and I'll request you all to join by 10:45. So first lecture will be from Professor V. Subramaniam. He's from CSR, senior scientist from the CSR lab, and he will be discussing some interesting aspects of structure-based drug design, where you will learn different types of different methodologies used in docking studies, molecular dynamics. All this part will discuss in theoretical aspects, and there will be break for 15 minutes, and after that we'll discuss the homology modeling, or we can say that the protein structure analysis theoretically and experimentally. And after lunch, we'll discuss the Autodoc software.
All right, the form is open, everyone. Please put the feedback. I'm going to close the form in next 15 minutes, 10 to 15 minutes. So please give the feedback so that we can record the attendance. And here is the, again, I'm putting the link for today's sessions so that if anyone wants to go back and check those sessions, you can do that.
So somebody is asking the form link again.
I'm putting that. Just give me a moment. Here is the form link again. Thank you everyone for joining today, and see you everyone tomorrow.
So we'll end this session, and the Google Form link will end in 10 minutes, deactivated in 10 minutes. So request you all to please give your feedback. Thank you. Thank you everyone for joining.
Shall we end the meeting, sir?
Yes, yes, sir.
Okay, all right, people. Have a nice evening, everyone. See you tomorrow. Bye bye.